Meeting started. I think we're waiting on a couple people. It sounds like, but we have a packed agenda today, so we want to keep moving forward. It's February 7th, February 8th at 9:34. We'll start our meeting. We'll start with introduction. I'm Erika Sawatzke, Chair of the Board. I'm a turkey producer from Kensington, Minnesota.
I'm Dean Compart. I'm a swine producer from Templin, Minnesota.
Jessica Fox, veterinarian from Southwest Minnesota.
Why don't we go around the table and then we'll get the folks in the back.
Okay. Brian Haight, Senior Veterinarian for the Board of Animal Health.
Maya Walker, Client Student Supervisor, Board of Animal Health.
Jopp, Assistant Director, Board of Animal Health.
Paige Brown, fourth year vet student at Exeter.
Carissa Allen, Data Manager for Board of Animal Health. Laura Brown, OAS, Board of Animal Health.
Erin Kreider, Communications Specialist at the board.
She's trying to fix the echo in case you hadn't figured it out. I'm Dr. Courtney Wheeler, Assistant Director with the board. Did you hear that 5 times?
Ashley Coles, Minnesota Turkey.
Marion Garcia, Executive Director with the board.
Addie Evans, Engelke, and Farm Service Program Administrator for the board.
Dr. Peggy Ann Hawkins is swine veterinarian, livestock veterinarian from Northfield.
Lucas Schultz from Minnesota Milk Producers Association.
Morning, Jill Ressler, CEO for Minnesota Pork.
Bill Lower, retired from the board.
I'm Dr. Scott Jones, I'm the veterinarian for Southwest Minnesota.
Scott Fier, President of the Minnesota Deer Farmers Association.
And William, Board of Directors, Deer Farm Association.
Jerry Campbell, Board of Directors for Minnesota Elk Breeders.
Secretary for the Minnesota.
Nikki Daly, Minnesota Agri Council.
Deb Boltwell's board member for Minnesota Department.
Mark Wolf, Board Member, Minnesota Deer Farm Association.
All big.
Do we have board member Jim online that he can introduce himself?
Is that what we need to do? Sounds like it.
We get inbox.
I see his name.
You see him on it. Is he talking? We can't hear him.
There was a circle around his name. I think he was talking.
Jim, are you muted? Could you unmute yourself and introduce yourself?
You see it? Michael.
Are we not able to hear him on our?
No.
That's
Michael's trying to help us troubleshoot it. Do you think it's because the microphone's hooked up, Aaron, to this?
It should come, the sound should come through the
Oh,
owl.
no, this is hooked up.
Right.
We do have our other board member, Alex, coming, but he'll be here about 9:45.
Well, do we want to circle back to Mr. Voss? I'm sorry, Mr. Voss, but then we have to establish try to troubleshoot this, and we'll come back to him. Is that okay with you?
Well, I could understand.
We're gonna go ahead and I'm gonna ask for, is there a motion to approve the minutes from December 6th? So we can keep moving.
I so move.
I second.
All in favor, please say aye.
Aye.
All opposed, same sign. Okay, motion carries. Is there an approve or a motion to approve the agenda for today?
I'll second.
Okay, there's been a motion and a second to approve the agenda for today. All in favor, say aye.
Aye.
All opposed, same sign. Okay, motion carries. Is it okay to move right into our first speaker so we stay on time?
As long as we can hear them.
Okay. Let's invite Nicholas Haley to speak. Hopefully we can hear you.
I can hear you. Can you hear me? Okay. Yeah, I went through and turned off everybody else's mic and that seems to have fixed the echo. Let me see if I can get my presentation uploaded here.
Fill the blanks. So Dr. Nick Haley is a veterinarian who also has a PhD in infectious diseases, and he specializes in genetic research in Cervidae species, and currently works for the College of Graduate Studies at Midwestern University. And he's joining us today to talk about genetic research in farmed white-tailed deer. And go ahead and add anything else you want about yourself, Dr. Haley.
Okay, thank you for the introduction. Sorry, I kind of glazed over that. Yeah, so I've been, can you all see the presentation on the screen?
Yeah. Yes.
Okay, and you can see the laser pointer and everything?
Yep.
Okay, perfect. So a little bit more about my background. I'm a DVM, graduated from Cornell in 2004, then went on to Colorado State University and did a residency in microbiology and a PhD focusing on CWD transmission and diagnostics. And since then, I've been working off and on with deer farmers around the United States and Canada, and on various projects, including live animal testing and more recently genetic research. And so I wanted to thank you all for inviting me. Thank Dr. Courtney Wheeler again for inviting me. And the focus of my presentation is going to be on one specific deer farm and the work that we've been doing with them. If we have enough time for questions afterwards, great. If not, feel free to shoot me an email at one of these email addresses and I'll get back to you. Let's see. So just making sure that slide moved on for you guys. Yes? No? Okay. So most of you are probably at least semi-aware of the chronic wasting disease situation in the United States and Canada. And as of the most recent surveys, the CWD has been found in 28 states, also New York, but it hasn't been reported in New York for over 10 years now. So it's kind of a 28 plus 1, 4 Canadian provinces now. CWD was first described in 1967, and it had been endemic in Colorado and Wyoming until about 1998, and then kind of has slowly been making its way across various parts of the United States. And so this map just, again, shows the most recent extent of the counties where CWD has been identified, both in free-ranging and captive deer. And just to kind of refresh your memory, this map is showing what the situation looked like 10 years ago. So quite a bit different in the past 10 years. And so the disease has expanded quite a bit. So more about the etiology of chronic wasting disease. So prion diseases, of which chronic wasting disease is one, result from a transmission and accumulation of a misfolded protein. And in the case of prion diseases, it's the prion protein. The process is similar to Alzheimer's and Parkinson's disease. We're just looking at different proteins that are misfolding and accumulating and eventually causing progressive neurologic disease and death. All animals have a normal version of the prion protein, but only some are susceptible to prion diseases. So most of you are probably familiar with cattle and mad cow disease, or BSE, and sheep scrapie, and CWD in deer and elk. And we also have some prion diseases in people, including CJD and Kuru. So how do prion diseases work? Well, as I mentioned, all animals, mammals, reptiles, amphibians have some version of a prion gene that makes a prion protein. So the prion gene is PRNP, and that protein directly modulates how susceptible that species, that animal, is to a prion disease. And so the prion protein is about 250 amino acids long, and there's some curved stretches and some straight stretches, and the infectious prion protein is, for all intents and purposes, the exact same sequence. It's just misfolded. And so how does the prion gene, the prion protein, directly modulate susceptibility? Well, if we have a single difference in amino acids, in the case of sheep, that animal can be more or less resistant to scrapie, for example. So the host prion sequence really makes a big difference in how susceptible they are to infection. And so some more background on that. So some research has been done on human susceptibility to Creutzfeldt-Jakob disease, variant Creutzfeldt-Jakob disease, and Kuru, and researchers have identified a couple of amino acids that play a really important role in susceptibility to those diseases. And many of you are probably familiar with sheep scrapie resistance, and there are a handful, 3 specific amino acids that are really focused on in building resistance to what's commonly called typical scrapie, so conventional scrapie, which is transmissible between sheep. And over the past 15 or so years, people in the United States and Europe and other parts of the world have been selectively breeding for sheep with what's known as the ARR genotype. So these letters here represent amino acids that are present at 136, 154, and 171. And by selectively breeding for sheep that have this particular set of amino acids there, it's led to the near elimination of classical scrapie in US flocks over the past 15 years. So we haven't had a case of scrapie in a couple years, and since 2017, there have been fewer than 5 flocks of sheep testing positive for scrapie. Overall, since 2003, the United States has seen a greater than 99% reduction in scrapie, just through a combination of things, but most importantly, the selective breeding of what's called ARR sheep. Now, what do we know about the situation in deer? Well, over the past 15 or 20 years, researchers have identified several different variants that whitetail deer carry. The problem is that none are completely protective against chronic wasting disease. And I have completely in parentheses here because we're not really sure whether completely will make will mean anything. We could have some that are partially protective, and that's still important. But I'll talk about that in a little bit. This figure here is from a paper that I published a few years ago. We looked at over 2,000 deer that had been depopulated, over 2,000 farm deer, and over half of those were CWD positive. And we basically looked at what the relative risk that different genotypes for the PRNP gene carried for those deer, whether they would develop CWD or not. And so what's on the x-axis here is disease stage. So we looked at the stage of disease, which I think is important because some genotypes move through the disease process slower. What's here on the y-axis is the odds of infection. So generally, genotypes that are more towards kind of the 0-0 point here have a lower risk of infection and also are more likely to be found in a lower disease stage. And what you can see is that the 96GG genotype here is what we consider the most susceptible. And unfortunately, it makes up most of the deer in farm deer herds and in wild deer herds, so 80% or more in some cases. And then different genotypic substitutions, so different animals carrying different alleles, like the 96S allele in combination with the 96G, or homozygous for the 96S allele, are going to be found in lower disease stages and less likely to be infected. The 96SS genotype frequency in nature is about 5%. So 5% of deer will be homozygous for that 96S genotype, relatively low susceptibility. So here's those animals here on that scale. And then what we don't know about is animals that have the 95H allele and are homozygous. And we do have some information on heterozygous, so animals that are 95H, 96G animals. And my suspicion is that homozygous animals are going to be way over here on this chart. But we just don't, they're very uncommon in nature and in farm deer until relatively recently, that is. And we don't have a lot of information on how susceptible they are. But my suspicion is that they're going to be even less susceptible than 96S homozygous animals. One important note here, and we'll talk about this towards the end, is that other genes, we talk about the prion gene, the PRNP gene as being important, but other genes can offer indirect modulation of CWD susceptibility. So how fast the prions traffic to the central nervous system, how rapidly they accumulate in the central nervous system. So there's all sorts of genes that might be affecting how this disease progresses. PRNP is just one, but there are others that we're learning more about recently. And so the project that Dr. Wheeler wanted me to discuss, we started this project in 2017. I was doing a lot of sequencing for deer breeders across the country, and a deer breeder in Wisconsin approached me about sequencing some of his breeding deer. And we also started looking at deer that had been harvested on their hunting properties. But basically, they have 2 breeding facilities and 1 hunting location that are considered CWD free. They have 2 hunting locations that at the time were CWD endemic, and 1 property had a 60% prevalence. So 6 out of 10 deer coming off the property were positive. The second property had a much lower prevalence, about 5%, but that increased over time, as I'll show you here in a little bit. So we started that project in October, and just with a thin thread of time, we had enough time to sequence his breeding deer and several prospective breeding bucks, and got their genotypes. And in 2018, we had our first fawns born using this selective breeding process, focusing on what we think are more resistant PRNP alleles. So pretty quick turnaround with the first fawns born. And subsequently, late 2018, I worked with the farmer to develop a 3-phase selective breeding plan to try to reduce CWD prevalence on those 2 hunting properties, and hopefully keep his breeding facilities in that other hunting location CWD free. And so we're doing our best to work on that. And so a little bit more about the project. So we've got 3 phases, and we've wrapped up phase 1 already, and that's basically getting the ball rolling on breeding deer towards more resistant PRNP alleles, and starting to introduce them onto the properties that are CWD endemic. We're in the midst of the second phase here, where we're getting a better handle on how resistant those alleles are, those genotypes are out in the field, out on those CWD endemic properties. And then phase 3 will come a little bit later, and I'll talk more about phase 2 and phase 3, what we plan to do in those those phases. So I wanted to start out with CWD surveillance. So we went and looked at all the deer that had been harvested prior to me starting work with this farm, and we've been doing all the sequencing since then. And what I hope you can see is that 96GG animals make up the bulk of the CWD positive animals, and we've got some mix, but we're focusing on trying to breed animals on this end of this chart here. And And to put that another way, on this side, we've got all the positive animals. On this side, we've got all the negative animals and what their genotypes are. And what I hope you can see is that 96 GG animals make up currently about 87% of all of the CWD positive deer that have come off of these hunting properties. And so to eliminate, if we can eliminate animals with these 96 GG genotypes in the herd, I think we can make a huge dent in CWD prevalence overall. I have warned the farmer and other people that I've talked to that, I don't think we're going to get rid of CWD, but I think we can make a huge dent in the prevalence and we won't see that 60% plus prevalence that he's had on that one endemic property. So where are we? So in 2022, just last year, the fawns born, none of them had that highly susceptible 96 GG genotype. And so this is looking at the genotypes of those fawns over time. This is 2017, the general population, the fawns that have been born in 2017, and then the fawns born in the first year that we started selective breeding. And here we are today. And so what we're trying to do is shift this bar graph over to the right. We want animals with more resistant genotypes, but we've certainly moved a long way in getting rid of this animal with this 96 GG genotype. When we look at specific alleles, so we've still got the 96G allele circulating in these animals, but by 2024, we hope that we won't have any fawns at all born with the 96G allele. We'll have what we think are more resistant alleles, and all fawns will have one of these more resistant alleles, and none will have this highly susceptible 96G allele. And by next year, we hope. And you can kind of see just the dramatic decrease in those susceptible animals over the past 5 to 6 years. Surveillance, this is more of a over time surveillance, and this is the two hunting properties where we know CWD is endemic. And so the first one had the really high prevalence when we started, about 60%. And that what we ended up doing in 2019, the owner had totally depopulated Site 1, which is about 400 acres of semi-wild property. So that was a tough thing to do, but he thinks in 2019 all the deer were removed off that property. So we're hoping that we're starting with a clean slate, and he started releasing animals onto that property with known genotypes. And so it shouldn't be too much of a surprise that we really cut down CWD prevalence there in 2019 after we depopulated the animals and we're starting to release them for known periods of time. So we started with a year they were on the property. 2021, last year, there were no positive animals on that property. So that's an improvement. And we're going to keep releasing animals onto this property with known genotypes for progressively longer periods of time. Site 2, what you can see is that, we started out with a low prevalence, and over time that creeped up. In between the 2019-2020 hunting season is when we started releasing animals with more resistant genotypes. And what I'm hoping to continue seeing is a decline in prevalence on that second site, now that we've got, kind of this injection of resistant animals onto the property, and hopefully they'll kind of spread those resistant genes among the animals that are there. But I think we've kind of blunted the spread of CWD, at least starting for the past couple of years now. So phase 2, this is the phase that we're in now. So we're going to continue our surveillance. I think the breeder, the owner, has got a really good handle on breeding, identifying animals to use as breeders and continue improving the resistance in his breeding herd. But we're going to continue focusing on genotypes, age, sex, disease stage, the time that those animals have spent on the endemic properties, to get a better handle on how long can an animal with X genotype exist on a CWD endemic property without getting CWD. So we can hopefully try to draw some cutoff points. Like we don't want to let animals live on the property for more than 2 years before they're harvested, for example. And we can, as time goes on, we'll get a better feel for progressively how long they can live on that property without becoming infected. So, again, we want to know, how does genotype affect longevity and disease progression? We want to get a better idea of animal-to-animal contact, what role that plays in disease transmission. So, on the first property, it was completely depopulated. So those animals, the only way they're going to get it is coming in contact with the environment. In the second property, we still have animals there that are maybe infected and transmitting it. So we've got animal-to-animal contact going on in that second property. So we can compare the two to get a better idea of, what's more important, environment or animal-to-animal contact. And then, like I mentioned, we can try to figure out whether there's a cutoff age that we can use to better manage the disease in farm deer herds. The third phase, we're going to focus on diagnostic improvements and whether prions adapt. So we want to know, can current tests, immunohistochemistry and ELISA, can those identify CWD in animals with these, otherwise rare prion gene alleles? We want to see if there's any evidence that CWD is adapting to those animals with those rare alleles. Like, it would be awful if, we did all this work and only to find out that CWD is now adapting to those animals and spreads just as easily. And we want to see whether prions recovered from those animals with what were rare alleles are different from those recovered from those highly susceptible animals. And I'll explain in a little bit, but basically what we want to know is, are we making things worse? Is it more zoonotic or is it more transmissible? We want to figure out if it's different. So in my time working on this project, I've heard of all sorts of arguments against selective breeding. And these are the arguments you may get from the public as pushback. And some people might suggest that the rare PRNP alleles are rare for a reason. That's a big one I've heard. Another one that I've heard is animals with these rare alleles are still susceptible to CWD. And I've heard CWD is hard to detect in these animals with rare prion alleles, and I'll provide rebuttals to all of these. Animals with rare PRNP alleles shed prions for longer periods. They're resistant, but they're still capable of being infected. They live longer and shed longer. And then there's the thought that CWD will evolve to infect animals with rare PRNP alleles and maybe become zoonotic. So some arguments, my arguments against, the rare PRNP alleles are rare for a reason. Well, what we found in one study a few years ago was that these alleles, the 95H, the 96S, they seem to be regionally more prevalent in some regions than others. And so what I think is that maybe it's more likely that they're recent mutations, and they're not rare because they're defective. They're rare because they're relatively recent and they haven't had a chance to expand. All of our fawning rates in this property, where we're breeding towards rare alleles, they match what you would predict. So we're not seeing fawns die in utero, for example. We're not seeing animals die at young ages because they're defective or anything like that. So I think that this argument really has no basis. And what several studies have seen in nature is that these rare alleles are becoming more common in areas with high prevalence CWD. So the 96S allele, for example, is becoming more common in central Wisconsin. The other argument, animals with rare prion alleles are still susceptible to CWD. One of the problems is that few studies have included animals with these rare alleles, so we don't really know much about them. Those few studies where animals with these rare alleles have been included have used really unnaturally high dose exposure. So like 10 grams of brain, which is probably 1 million times more than an animal would see in nature. And so, it's hard to interpret that when they're getting overwhelmed with infectious material. Same thing goes with the field studies. So those field studies that we looked at where some of these rare alleles were present, were really high prevalence areas. These were farms where they had 500 deer on 40 acres, for example. And so, the CWD prevalence was incredibly high. And so we're not really sure what a natural dose would do to these animals. Another argument is CWD is harder to detect in animals with rare prion alleles. And that's true. But one of the reasons is because fewer are getting infected. So, we have to spend more effort trying to find those animals that are infected. And as I mentioned, in phase 3, we're going to continue working on the tests to make sure that the tests that we have now adequately identify positive animals with these rare alleles. And so we'll be working with the USDA to try to make sure that those tests are still effective. The argument that animals with these alleles shed prions for longer periods, they live longer, so they shed longer. Well, you know, I've heard the typhoid Mary argument, and you know, one question that I always have is what is the importance of a typhoid Mary in a group of a whole bunch of typhoid Marys? You know, typhoid Mary was a woman who was resistant. I've forgotten the disease that she had now, but she was resistant. She was asymptomatic, but she would infect other people. But if everybody else was a typhoid Mary and they were less likely to be infected, you know, what's the relevance of having these resistant animals where every other animal is resistant? We don't really know how relevant that is in herds with less susceptible alleles. And on top of that, you know, like I mentioned, we can target harvest and culling ages to make sure that animals aren't living longer than 4 years, for example. So they're, you know, no matter what, they hit a point where they're no longer shedding. The last one is CWD will involve to infect animals with rare alleles. We have some ongoing studies to look at prevalence changes among these animals with rare alleles. We have a mixed background, so animals have, you know, we're going to kind of shift what the prion sees, so it will hopefully minimize the risk that it will adapt. And then we have planned studies with the USDA and the NIH to look at whether other species may be more susceptible now that we've got prions that are in animals with these rare alleles. So is it more infectious to people? Is it more infectious to elk or other mammals, for example? So the last thing that I wanted to talk about really quickly, just to kind of wrap things up. So my work kind of started based on what we knew about prions at the time and what was being done with sheep. Since then, Dr. Chris Seabury with Texas A&M has been working on genome-wide association studies, or GWASs. And these look at the prion gene, but they look at, no joke, tens of thousands of other genes that deer have to try to figure out, are there any other genes that might be affecting how susceptible the animals are? And he's found quite a few. And what this genome-wide association studies gives him is estimated breeding values for individual animals. So he can assign a breeding value to animals, and you can use those breeding values to pick animals that are more or less resistant. And it gives a numerical value to those animals. And over time, that breeding value data is refined as he gets more data, so he can put more accurate numbers on those breeding values. One of the drawbacks is, so you get more and better data, but it's a little bit more costly than you know, prion genotyping, which runs me about $15 a sample. His genome studies will, you know, run, you know, five times more than that. But I think that the value is worth it. It's not as immediately rewarding. So, you know, we're getting a turnaround time of a week and giving, you know, people are breeding, and, you know, in 5 years, we've completely eliminated animals that are homozygous for the 96G allele. The problem with the genome-wide studies is that there are, again, tens of thousands of genes involved. Those genes could play a role in Alzheimer's and Parkinson's pathogenesis, so they're really important to consider, but it's really hard. I know that farmers will get frustrated when they breed 2 deer with what looks like good breeding values and get a fawn with a bad breeding value, but that's possible because there's tens of thousands of genes involved, and depending on how they're passed on to the fawn, you know, it can be frustrating. But I think in general, this process that Dr. Seabury has is going to be much more rewarding in the long run than focusing on a single gene. And I think that deer farmers should be focusing on this where they have time. A lot of deer farmers don't have time, which is unfortunate, but that's kind of the way things are. So I wanted to quickly thank the breeding farm and hunting ranch owner operator. He's been great to work with. Most of you probably know him. I couldn't ask for a better collaborator on this project. Drs. Amy Horn-Deltzer and Darlene Conkle with DAC in Wisconsin. I know that you guys are probably familiar with being close neighbors with Wisconsin with their state veterinarians. I've had several veterinary students that have helped on this project, and I wanted to thank Dr. Wheeler and you guys with the Board of Animal Health for inviting me to talk. And I've got some references here just to kind of reference, all the points that I made, the arguments against the arguments. So, yeah, with that, I'll take any questions if we have time.
We have a question in the back.
Okay.
Dr. Scott Josephson, Tri-County Clinic in Taunton, Minnesota. I spend a lot of time working with reproductive programs on deer farms throughout the Midwest. I've got two questions. The first being, you discussed the fact that we're selecting for these alleles, and I would say that in the number one selection that we deal with deer breeding farms is resistance. That's their number one criteria, is how resistant.
Oh, can you guys? I lost the last bit of your question.
I was just explaining the number one criteria that our breeding farms use when they're selecting bucks to use semen for artificial insemination is resistant identification. So our guys the people I work for don't use bucks that don't have resistant alleles, is what I'm trying to say. But my first question is, you discuss incomplete resistance, and I'm wondering, and I've thought this for a while, is there an additive effect to the resistant alleles? So for instance, if you get a 95H and or a 96SS and you stack on the resistant alleles in an individual animal, does that add to the resistance that we see in these animals? Is there any evidence of that, or have you looked at that at all?
Okay, so I'll answer your first question, but I'm guessing you have another one. So one of the problems is that these alleles are mutually exclusive. So like you see with sheep, where they can have an ARH, they can have an ARR, they can have a QRH. We don't have that in deer, where you can't have an animal that has a 95H and a 96S on the same allele. You can have a 95H allele and a 96S allele in the same animal. So you can't technically stack them, but if you have an animal that's heterozygous for 95H and 96S, in theory, they'll be more resistant than an animal that's 96S, 96S, if that makes sense. Yeah, so there's some stacking, but because all these mutations aren't on the same gene, you can't, kind of exponentially increase resistance that way. We're looking at individual alleles. I hope that makes sense, but if not, you can shoot me an email and I'll try to explain it better. Did you have another question?
Question is, in talking with the clients of ours that we work with, when they establish higher levels of resistance in groups of for instance, young breeding does or so forth, I hear comments from these people that the more resistant animals they have within their herd, the healthier and more productive those animals seem to be. So this is a question that has no answer, but I wonder if we're learning more and more about this, but as we select for these resistant alleles and as we create more desirable breeding values, are we also, without knowing it, selecting better general immunity within those animals so they aren't susceptible to other issues that may cause them to be less productive? That's just an observation from the field and comments that I've gotten back from producers that I work with. Do you have any comment on that whatsoever?
Well, I agree that that's kind of subjective and anecdotal, and I'm really glad they're saying that and not saying that those animals look worse, right? Because that gives them motivation to do that. I mean, I haven't heard anything anecdotally from this farm that I'm working with in Wisconsin about that, other than they're not. He can't tell them apart. So I think, generally that's good. At least we're not making things worse. But if you're familiar with breeding dairy cattle, you know that there is a possibility that you could screw something up and introduce a gene that could have widespread problems. But hopefully we're smart enough to avoid that. but we're doing our best, I guess. Yeah. Any other questions?
Dr. Haley,
It's hard to hear. I don't know if it's the mic.
speak up. So my questions were about your phase 3 that you're entering into now. You mentioned validating current testing for CWD, which I think is great. Are you also looking at incorporating some of the new testing that's out there? So in Minnesota specifically, our legislators are focusing on RT Quick. Have you been approached?
So I've been a long time proponent of RT Quick, and that's definitely something that we'll look at. I personally think if we have to make modifications, it will be easier with RT Quick than it will be with IHC. IHC, we might have to find completely new antibodies and completely new validation steps, but with RT Quick, I think, at least what we know so far, it doesn't really care what genotype the animal is when it's amplifying prion. So I think it'll be much easier to do that than with IHC. Yeah. And you might have to speak up.
Sorry, in addition to testing the animals, are you also looking at testing fecal samples, soil, anything like that on this farm?
We haven't been. I mean, we do have a paper from a few years ago, but there's no real ongoing work that looks at fecal samples or environment. I mean, I'd be happy if people are interested to talk to the owner and see what he's up for a lot of things, and so like I said, he's been great to work with, and he's been very proactive, and he wants to see a solution to CWD as much as anybody on the wildlife side of things.
And then one last question, if you can still hear me, for that
Yep.
phase 3.
I lost you a bit.
I'm quiet.
I'll turn you up.
All right, so my question had to do with susceptibility to other species, and you said in that phase 3, that's something that you're looking at. So is this particular farmer looking at putting other species on that CWD endemic property?
No, what I think we'll do is, so he sends me lymph nodes from every animal that's harvested, and I've got a freezer full of them, and we'll work with people in Calgary, for example, people in the US with the USDA, for example, and look at if they want to do raccoons and pigs and mice and whatnot, that's fine. I mean, we've got all these isolates and they're frozen back, and anybody who wants to look at whether certain species are more susceptible or less susceptible now to these new prions, then that's fine. Yeah.
Can you hear me okay?
Maybe a little bit louder.
I wonder if it's a connection issue, maybe. So I wish that I would have gone before Dr. Josephson and Dr. Wheeler, because mine are going to sound pretty basic. But just to clarify, so you guys kind of went this direction because this was what was done in Scrapie and it was successful. Is that correct?
Yes.
And have other prion diseases adapted, such as Scrapie? Have you seen that where there have been the adaptations people were worried about?
We haven't, at least in natural settings. In experimental settings, what can happen as prions adapt to a new species, like they were taking Scrapie and inoculating hamsters and stuff, and what they found was that when they inoculated hamsters, other species became susceptible that weren't directly to Scrapie. So when you put Scrapie in a hamster, other species are now susceptible that weren't directly susceptible to Scrapie. So experimentally, that idea is there. I think it's a concern, but it's like I mentioned, this is something that's happening in nature where these genotypes are becoming more common in areas where CWD is endemic, and so I think we need to know whether or not it's a possibility because it's happening in central Wisconsin right now, whether we want it to or not. And so it's important to know, if we can get ahead of it with more controlled studies, then we'll at least be better prepared. I can't hear you now.
Maybe I can type it.
I still can't hear you. I heard you for a second. I'm pressing my head up to the computer, but it's not working.
We'll type it into the chat.
Okay.
As she's typing her question, Alex, can we have you introduce yourself?
Alex Beatty, member of the Shock Committee, Lockton Sioux Community, and cattle producer.
Dr. Fox, did you want to know in farmed animals or wild animals or both?
Oh, just in general, are there different variants of CWD?
Okay. Yes. So great question. So there are. It's a lot of it that has been identified in the lab. Where they take CWD isolates from different animals and put them into another species, and what they get looks different. Like they inoculate a whole bunch of mice, and one group of mice looks different than the other group. So best estimates suggest there's at least 3 different CWD variants, but we really have no idea what that means. We just know that they're different. We don't know that one is more infectious than the other. We don't know that one causes longer periods of infection. So it's really, we don't know. We know they exist. We don't know how important they are.
At that meeting yesterday, weren't they talking about 7 or 10 variants
Yeah, Peter Larson's group,
of CWD?
right.
Did you hear that, Cole?
So I'll just say that there are at least 3 papers that talk about multiple different strains, quote unquote. I don't know. Some of those papers could be talking about the same strain, and they don't know because their approaches are different. So at minimum, there's 3 different strains, but if you counted all of the ones from those 3 different papers, there's 7 to 10, but some of them could be the same strain and we just don't know.
I think, Dr. Haley, can you hear me?
I can hear you, yes.
I think there's also been some debate on spontaneous cases as well, right?
Yeah. The problem is we wouldn't know a spontaneous case if it hit us in the face because we've got so many transmitted cases, right? It's not like the BSE epidemic where now that they've gotten rid of classical BSE, they can really easily identify the spontaneous ones. It might be 1 in a million, but we've got a million transmitted ones to sift through. So.
In that preserve, you said they depopped it in 2019.
Yeah.
So I'm just trying to wrap my mind around. So they depop it in the spring and then had deer on the landscape again in the fall, or how long? And to give us an idea, was it 3 years that deer were in there before the ones that were harvested this past fall?
So there was a relatively short period of time between when it was depopped and when they introduced animals. So I wouldn't expect there to have been any environmental degradation, but we, in theory, eliminated any contact transmission. The only way the ones that are being put on there now are going to get it is through coming in contact with the environment. But it was depopulated in the winter of 2019, going into 2020. And then he started releasing animals onto that property within a few months to be harvested in the fall. So that first year they were there on premises for 6 to 8 months. And then he put a few more on, and they were there for like 18 months. And so the thought is that each year some of them are going to be on there for a little bit longer. And so we've got when they were put on the property and when they were harvested and their genotypes, and we can figure out exactly how long it takes them to show up lymph node positive or brain positive or whatever.
The essence of time, I would like to move on to the next speaker, but
Perfect.
if there are any questions from anybody, please feel free to reach out to Dr. Haley. We can probably get his contact info from Courtney.
Yep.
Yeah. Thank you very much for your time and all your information, Dr. Haley. We appreciate it.
Yeah, thank you guys so much for having me.
And I see we do have Dr. Molgaard. Go ahead.
Thank you very much. Can you hear me okay? Can everyone hear me?
Yes.
Yes.
Okay, and can you see my slides?
Yes.
Okay, great. So it's a pleasure to be with you today to give a brief update from the College of Veterinary Medicine. I just wanted to share with you some kind of brief updates from around the college. Won't be comprehensive at all, but these are some photos of some 3D models that our MinPro Prion Research and Outreach team created. A team from the USDA was visiting the college a couple of weeks ago, or actually it was maybe just last week, and they will be licensing the access to use these models for training of their workforce for to use. Right now what they're using is actually just photocopies of drawings, and these 3D models will be much more effective to train food safety and inspection services staff across the country. These are models of deer heads, but there are also models of bovine heads that can be used for training to find lymph nodes and brainstem, etc. And you can see these products if you go to that website, or if you just Google MinPro available products, you can see these 3D models and more models, but they're pretty neat. They were made from high resolution scans of actual heads. Another update is about the Center for Disease Control funded Pathogen Genomics Center of Excellence. We are finalizing agreements for funding from the CDC, and this is one of 5 such centers in the country. So the Department of Health is the lead agency for the Minnesota Center, and with this funding, Dr. Tim Johnson will use MDH data and isolates to test the hypothesis that our predictive tools can accurately predict high-risk strains that result in human outbreaks. The Minnesota project will also work in other areas, including SARS-CoV-2 and antimicrobial resistance. The primary pathogen that Dr. Johnson is working with is Salmonella. I've mentioned before that we're in the middle of, or we were in the middle of strategic planning, and now we're towards the end of strategic planning. And I won't go into most of the details, but I want you to notice that at the bottom of this arrow, you can see that our 3 primary pillars are engagement of people, infrastructure, and partnerships and constituents. I mentioned that because I'm going to be talking about some of that in the remaining slides. So within engagement of people is our workforce challenges. So our college, like other colleges of veterinary medicine, is facing a really crisis of clinical faculty workforce. We have workforce shortages in other areas, as I'm sure all of you do, but in our clinical faculty workforce, this is becoming really a challenge, not just in delivering our service mission, our clinical mission, but also in delivering our educational mission. So several key programs in our college, a couple examples, our medical imaging and surgery, are at serious risk due to our inability to recruit and hire qualified faculty members. And so when we can't hire faculty members, when we don't have faculty members, we not only can't provide service and conduct research, but we can't train the next generation of specialists and we put at risk even the training of the next generation of veterinarians. So part of this, and it's only a piece of the puzzle, is the Practice Act. The Minnesota Practice Act puts us at a competitive disadvantage because of some limitations that limit our ability to recruit faculty who've been trained in international veterinary schools. So what we would like to do is to change the Practice Act to emulate language from some other states. An example is Kansas, to create an institutional license that gives the college more discretion. And that would create, again, more discretion to have that institutional license. And once, if those faculty were to leave the university, they would lose that institutional license and have to get a regular license like any other veterinarian. So what are the risks of not doing this? Again, we can't train people. We can't perform our service mission. This is also some of the strategies that colleges who are facing similar kinds of challenges are having to resort to is outsourcing more of the education, like hiring private companies like VetCT to teach radiology, or outsourcing more of our education through the distributed model of education. Those of you that are familiar with some of the newer veterinary schools, that rather than having a teaching hospital, they send students out for their clinical year. So legislation has been introduced in the Minnesota House, and we're waiting for a hearing on that. And we have a meeting with the Minnesota Board of Veterinary Medicine next week to talk about this. We've had phone conversations, but we'll be having a meeting with the board next week, and the MVMA is in support of this. More on workforce. Our rural veterinary workforce continues to be a challenge. And one of the strategies to support the rural veterinary workforce is the Rural Veterinarian Loan Repayment Program. The application process for that closed last week. And just by way of reminder that there's funding available in the amount of $375,000 each year for 5 veterinarians or graduating veterinarians who will practice in rural areas and serve food animals at least half of the time. Other strategies that we have put in place to support the rural workforce are VetFast, our Food Animal Scholars Track program, and the newer program with SDSU. We are continuing to invest in infrastructure, part of our strategic plan. And this is just one example of some infrastructure investments, or 2 examples. Our transformational learning corridor is some remodeling that we're doing on the 2nd floor for students. And then this is our interventional radiology suite for clinical service. There are others, but there's a couple of pictures there to show you some of the investments that we're making. And these investments are typically funded through a variety of sources, including philanthropy and partnerships, as well as collegiate funds. And then just some announcements of upcoming events. The White Coat Ceremony is coming up April 22nd. Commencement is coming up in May. And then just a reminder that we are celebrating our 75th anniversary this year, and that will be happening in May. So thank you very much, and I'm happy to answer any questions.
All right, thank you, Dr. Molgaard. We appreciate your update.
Thank you.
Say, would we be able, do we have our technology figured out that board member Jim Voss could introduce himself?
He just has to unmute.
You what?
He just has to unmute himself.
Okay. Hey, Jim, could you unmute yourself so you could introduce yourself before we move on?
The vocal is not very good, so I assume you asked me to introduce myself.
I did.
Okay, board member Jim Voss.
Hey, could you tell us one more thing about yourself?
Oh, I live in Fillmore County. I'm a fourth generation farmer. I've been farming there for many decades. And I had multiple livestock, beef, hogs, and dairy originally. So I'm honored to be on this board.
Thank you, Jim. Appreciate that. We're gonna, to stay on time, we're gonna do a little bit of shifting. I'm gonna bump Dr. Garcia down. Do we have Michelle available for her update?
She had to step away for a meeting.
She did.
I did message her and see if she
Okay.
can.
Would you want to start your cattle update?
So Michael sent me a message, and that the issue with Teams and Q&A is if you just keep speaking, it'll eventually pick it up. It loses the first couple seconds, so just stick with it.
There's a delay.
Yeah.
So everybody needs to start their question with la la la.
Yeah, random fill, yeah.
Okay, there we go. Everything's delayed today, right? Wait for. All right, good morning, everybody. I know we're kind of in a hurry today, so I'll try to be as brief as possible, but most of you know I do like to talk. I do it often. So I wanted to talk a little bit about cattle programs because we haven't visited cattle programs in quite some time at these board meetings. So I'm pretty excited to talk about cattle today. Just a couple of announcements. We actually have a new cattle programs director starting with us on February 27th. Dr. Katie Corneal. Katie, were you able to make it today? She was going to try to jump on.
Yeah, I'm listening in. I'm here if you can hear me.
Want to tell the board a little bit about yourself, please?
Yeah, absolutely. Hello, Dr. Katie Corneal. I grew up in Minnesota, did my undergraduate in chemistry out in Pennsylvania, moved back here for my doctorate of veterinary medicine at University of Minnesota, where I graduated in May of 2020. Out of that, I joined the USDA Food Safety Inspection Program, where I have been serving as a supervisory public health veterinarian in the St. Cloud circuit. So while that's been a good experience, I am super excited for this transition and to work with what seems like a really amazing team and to see and work with you all in person.
So much, appreciate that. Look forward to seeing you in a couple weeks.
Thanks.
And to her comment about an amazing team, I just wanted to recognize our cattle staff in our office in St. Paul. Becca White is our program administrator, and she did a lot of the research I'm going to present today on our transition to electronic certificates of veterinary inspection. Also want to recognize Nancy Reek and Avery Burgum for the great work that they do for the cattle program and moving us forward into this century. All right, so to get started, just a little bit about the cattle programs in Minnesota. So as you're aware, like with all of our programs, we partner with state and federal agencies, producers and stakeholders to detect the introduction and mitigate spread of diseases. I'm just going to touch base on a few cattle diseases that we look at routinely, but know that there are many that I'm not going to address today, including foreign animal diseases. So perhaps at the next board meeting, we can have some time to talk about our response to foreign animal diseases, not only in poultry, but in other species, including cattle. Bovine tuberculosis, as you're all aware, is contagious infectious communicable disease of animals caused by Mycobacterium bovis. An interesting fact, TB was once the most prevalent infectious disease of cattle and swine in the US and caused more losses to US farmers in the early part of the 20th century than all other infectious disease combined. So we kind of have forgotten about TB in Minnesota, thank goodness. But just, we want to keep it on our radar because obviously it's very detrimental to the cattle industry. Thanks to the implementation of a nationwide TB eradication program, which started in 1917. Anybody in this room alive then? No? The majority of states have effectively eradicated TB, including Minnesota, and we actually eradicated it twice. And I'm looking at Paul because he was there the second time. So in 1971 and then again in 2011. And I will tell you that I worked in an administrative position for the board in 2005 during our last TB outbreak. And people in this room, people that work for our agency, our field staff, everyone just did a great job with that response. And I'm very confident that if the need should arise, we can do that again in the future. Nationwide, the prevalence of TB is very low. Like I said, most states have times per 1 million cattle herds annually, and produce diseases we talk about frequently at these meetings. There's a live animal test for bovine tuberculosis. So that's really been the reason we've been so effective in mitigating the spread of this disease.
That was weird.
Is that Lisa? Gotta mute Lisa. All right, so again, Minnesota remains bovine tuberculosis and brucellosis free. And this is thanks to our more than 500 accredited veterinarians who conduct this disease surveillance across the state. So diagnosing TB in live animals is accomplished using a testing technique that is an intradermal injection of tuberculin in the caudal fold of that animal, so in the tail. Minnesota veterinarians are required to be certified to test any species in the state for tuberculosis, and our folks provide training to veterinarians because there are some differences in how different species are tested. In fiscal year 2022, Minnesota veterinarians screened more than 5,000 cattle and bison for bovine TB using the caudal fold test. And then we had 20 responders, which required additional testing by our regulatory field veterinarians. And fortunately, everything was confirmed to be negative. Since 1934, USDA and state animal health officials have worked on eradicating brucellosis. And I kind of joke about this, but when I took on the responsibilities of this program, it was really hard to find anybody with sufficient knowledge about brucellosis because it's been gone so long from the United States. And I attended a USDA training and they actually had to bring in veterinarians from retirement to talk about it because they had actually had an experience with it. So fortunately, I've never actually seen a case of brucellosis in cattle in any state. Minnesota was certified brucellosis free in 1985, and we maintain that status today. And really, brucellosis now is primarily found in the Greater Yellowstone area of Idaho, Montana, and Wyoming because it's endemic in wild elk and bison. So we continue surveillance for brucellosis. Positive test results are reportable to the board, and we follow up on these samples, conduct whole herd tests of animals if necessary, or additional surveillance for brucellosis. And a significant number of cattle are still vaccinated against Brucella abortus. Keep in mind that animals over 12 months of age can only be vaccinated with approval from the board. We did have a couple of these this year, not too many. And in fiscal year 2022, 116 cattle were tested, all negative for brucellosis, and more than 13,000 cattle were vaccinated.
Courtney, quick question.
You bet.
Why would you vaccinate, or what would cause you to be one of those that vaccinate if it's not done routinely?
Yeah, so primarily it's for movement requirements. That's really why people are doing it. Yeah, Alex, if you want to comment.
Well, I'd say a lot of the cattle shows that we go to, we have to, vaccinate for it, so.
Yeah. And one thing I want to note, because we've gotten this question a lot lately, is it used to be when you vaccinated cattle against brucellosis, you had to put in this orange tag that you see in this picture. Now you can put in any official identification tag. It doesn't have to be the orange tag, but they still do require that tattoo. Joni's disease, contagious, chronic, eventually fatal, affects the small intestines of ruminants, not pretty, caused by Mycobacterium avian, subspecies paratuberculosis, and it's really hardy in the environment, unfortunately. But the biggest concern for dairy farmers, specifically in Joni's, is that it costs lots of money. So they estimate animals infected with Joni's average loss of $40 per cow, up to $227 per animal, which I'm looking at Al because he knows that's a pretty big deal. That's a lot of money. So primarily these losses come from reduced milking, early culling, and poor body condition scores. We currently have 19 Minnesota dairy herds that actively participate in our voluntary Joni's disease control program, which requires them to test their animals annually. This testing is primarily conducted by the Minnesota Dairy Herd Improvement Association, and then they share those test results with us. So they actually have technicians that will go out to these farms and collect these samples, and then they run them at the DHI lab.
Why only 19? That doesn't seem like very high number.
I think because we used to have funding available for this testing, we no longer do, so it's out of pocket for all these folks. We've also, what we used to provide on the state side and on the federal side was a Joni's risk assessment, because we found that in addition to testing and culling, there are a number of things that producers can do related to biosecurity, et cetera, to limit the spread of Joni's or limit the introduction of Joni's. So I think a lot of people, and Alex can chime in anytime he wants here, but I think a lot of our producers just do such a good job with management that it's become less of a problem. Yeah, absolutely.
It's a lot cheaper to manage through it, and that doesn't, 19 doesn't count how many will test. People will say, oh, I think she's a suspect cow, and so that once a month test, you'll test her and maybe cull her or say, oh, she's negative, so we'll keep her.
Sure.
So 19 is not the full testing going on.
That's a good point. And there are a number of producers that don't participate in this program, but then will test a subset of their animals just to screen for the disease. They do pooled testing. So thank you for that comment.
And those 19 herds, were they infected beforehand? Is that why
they participate?
Yeah.
That's a good question. And I don't know the answer to that. I can tell you that the point of them participating now is that they don't have Joni's.
Yeah. I think when that program popped up, it was basically endemic. I mean, everybody had some semblance of it. And so you might have joined because you thought you had zero, or you might have joined because you thought you had a lot, or just for fun. But I think everybody had it. And the vets would know more than me, but.
No, I think that's probably true, sadly. Touch base a little bit on anthrax, naturally occurring disease, and it's not spread animal to animal. Rather, anthrax spores survive in the soil and are ingested by livestock when they graze on pasture. And then the spores become active once they're ingested. All warm-blooded animals are susceptible to anthrax, but cattle, sheep, and goats are the most commonly affected. And in rare cases now, humans can contract anthrax after handling or eating infected animal products. And probably a number of people in this room has been vaccinated against anthrax. We have. Since 2000, all positive anthrax cases in the state of Minnesota have been confined to the northwestern part of the state. So many thanks to Dr. Cater up in that area, because he's responded to most of our anthrax cases in the history of the board. And the last reported case of anthrax in Minnesota was in 2013.
What are the symptoms of anthrax?
Yeah, death. And lack of rigor, so no rigor mortis.
And some places it typically shows up when it gets very dry. It's like water levels go down, or isn't that?
Yeah, so
necessarily the case?
Erik, you can help me out with this one because you probably, have been more privy to anthrax than I have. But my understanding is it has to do with those areas that are arid then getting flooded, causing those spores to resurface in the soil and become available for those animals to ingest.
And I'll say the thing, anthrax in pigs, basically the pig dies and bloats very fast. And so, within hours, they're bloated and no rigor and everything. Do not cut that animal open.
Thank you for reminding everybody.
It's in the blood. Do not cut that animal open and do everything you can to just call the state veterinary.
Good job. Thank you. Appreciate that. All right, and really what I wanted to talk about today and hopefully get some support on is our efforts for animal disease traceability, specifically looking at moving to electronic certificates of veterinary inspection. So as you know, animal disease traceability is important for all the work we do. And how producers can help is by applying official identification and keeping accurate records, which for some people is harder than it sounds. So we still have some work to do. The board utilizes cooperative agreement funding every year to encourage the use of electronic ID and streamline record keeping, and I'll talk a little bit more about that as we move forward. So as you are all aware, official identification is mandatory for most cattle in Minnesota with a few exceptions, as it is for most species. And USDA has teamed up with state animal health officials to recognize certain ear tags that use a unique alphanumeric system. So there are certain tags that are approved for use and certain tags that are not. Dr. Schwabenlander, when she worked for us, did just a really great job of developing these resources, these guidance sheets on which official identification is approved in which species. And we continue to use them because I just think they're really well done and give a really good explanation and have good graphics. The point of this official identification allows us to trace individual animals that have been exposed and may be infected with a contagious disease. So that's why it's so important. In addition to official identification, we need producers, livestock markets, exhibitions, sales, and veterinarians to keep detailed, accurate records of all cattle that are imported, exported, and moved within the state of Minnesota. And I can feel our field staff on this call rolling their eyes because it is surprising that the number of producers, markets, even veterinarians that do not keep accurate records. So something really important if you're having a casual conversation with somebody at dinner and they're a livestock producer, you might want to say, hey, have you made any good records lately? That's really important. We would appreciate you educating people and absolutely send them our way if they have questions. So I'm just going to illustrate the importance of official identification through a pretty cheeky example here, and it's going to be brief, so don't get too worried, but I want you to note that in this scenario, I did not change the name of this cow to protect her identity because that would defeat the entire purpose of the ADT program. So this is Clara, named after my daughter. She is a 3-year-old Holstein cow who was sold at a Minnesota market in May 2022. And as you can see, she does not have official identification. Well, she could have a tattoo, but she doesn't. Okay. Cow in the same kind as Clara at this livestock market tested positive for bovine tuberculosis. So the board dispatched one of our trained investigators to make sure Clara got tested for tuberculosis. The investigator found that Clara was loaded onto a trailer that same day and commingled with 10 other 3 to 5-year-old Holstein cows who actually looked very similar in appearance to Clara. You can imagine. 2 cows were sold to Minnesota Farm A, where they were commingled with 10 more Claras and sold to 5 different farms and so on. So you can see how where this is going. So the biggest question there is what happened to Clara? And the answer is we don't necessarily know. We may never actually find Clara. So what happens in instances like this, and unfortunately all too common, is because we can't find Clara, an awful lot of animals get tested for tuberculosis. State animal health officials, veterinarians, livestock managers spend an awful lot of time tracing these animals down, and we spend a lot of money that we wouldn't need to spend if this animal had been officially identified. In an effort to assist farmers and stakeholders in improving ADT, USDA is working towards nationwide utilization of RFID tags in cattle starting. So if you're not familiar with RFID tags, it's an electronic tag. This picture has a button, but you can also get a dangle for a management tag. Alex, I don't know if you use any of these in your place. Okay. Yeah. And then they can actually be tracked, I keep moving this way, through a wand reader or panel readers to record the identification number of the animal. You'll notice that these numbers are 15 digits long. So the fact that they're electronic is exceptionally important in the fact that when these get written down, you can see how someone could easily misconstrue one of these numbers or make a mistake. So the Minnesota Board of and we want to give them away because we don't want to use them. So please send people our way if they're interested in tags and trying them out. We also have a number of ones for reading these tags that we're happy to loan out to folks. And then USDA also has heifers, and we get those requests every single day. And Kelly Neisen with USDA, places those orders for us and make sure that those tags get to those farmers. Another important tool for animal disease traceability is a certificate of veterinary inspection. I'm not going to go into great detail because I think everyone in this room understands what a CVI is and why it's important. But what I do want to talk about is the difference between electronic certificates of veterinary inspection and paper CVIs and why we want to move towards the electronic version. So you can see here, based on this graph, this is the number of electronic versus paper CVIs by species. And Dean, look at your swine folks.
Okay, where now?
See them?
This one.
Okay. They are just doing a great job, right?
Oh, yeah. Yeah.
I mean, they're just rocking it. Some of our other groups need a little bit of work, right? To get them on board with using these electronic forms. So in 2022, and again, thanks to Becca Weitz for all this great information, all this background research she did to give me these numbers, but we processed 22,477 export CVIs. And I got to tell you, that's a feat for these folks. I'm really proud of the staff in our office because it's a lot of work to process these. 9,000 were paper and 12,000 were electronic. So why do we want to switch to electronic versus paper certificates of veterinary inspection? And it really comes down to money, time, and resources. So Becca just did a breakdown here for costs associated with paper CVIs versus electronic CVIs, and you can see there's a pretty huge difference in how much we're spending on paper CVIs versus the electronic format. In addition, people hours. Our people spend tons of time processing paper CVI. So we have a system that we use called CVI Central. So if you have specific questions about it, she's the person to ask. But when that system receives an electronic certificate of veterinary inspection, it can recognize most of the information on that form and automatically import it for us, including identification numbers for those animals. So that versus paper CVI, where our people have to hand enter all that information. And other states have already done this and done a really, really good job. As you can see, Montana has 99% use of electronic CVIs by their veterinarians. Indiana and Georgia aren't too far behind them. And I think Wisconsin's coming out with some more numbers and they're doing a really good job too. So we're looking at how do we incentivize our veterinarians to use these other than saves everybody time, money, and energy. And so some other states have had some incentive programs where they say, we're going to charge you for certificates of veterinary inspection. We're only going to supply you with so many and then you don't get any more. And also we're going to provide you with what we refer to as goodies, an incentive for using them. RFID tags and ones, right, to help you make that transition, and you can provide those to one of your clients and work through that process. So in conclusion, compared to states with comparable or greater numbers of animals exported, the rate of eCVI use in Minnesota veterinarians is considerably lower, except for our swine folks. eCVIs are far more cost effective and efficient than paper CVIs. Paper CVIs are becoming antiquated technology, and we need to update our policy so that we can provide the most cost effective and time saving ADT efforts to people and animals in the state of Minnesota. So really to provide a better service to all of our stakeholders. That being said, we came up with a timeline for phasing out paper CVIs and rolling in electronic CVIs. This is actually a better graphic of what that looks like. And I think the gentleman in the middle is actually a veterinarian. I think he really is. So this year we plan to educate notified veterinarians, livestock concentration points, the public, that we plan on transitioning to electronic. And then next year we'll work further on the implementation of those education and support to make sure everybody has the resources and tools they need to actually access electronic certificates and utilize them. And then in 2025, we're going to stop accepting paper CVIs. So that's where we're headed. Questions or comments? Dr. Hopkins.
As a swine veterinarian, I've probably written 5 paper CVIs and hundreds of electronic. In the swine industry, we move pigs.
Yep.
and we have to know where they're at. And there's a way that you can have group pigs with tattoos, so you don't really have to put a tag in everybody's ear. It's just fantastic to do the electronic. There's also a company that has developed who will help with doing electronic Coggins tests in horses. Horses is always as electronic now. You can't do it by paper anymore either. Is that right? Is that from the Board of Animal Health?
I think it's still allowed paper Coggins. That would be very many,
But you still allow paper?
There's not many out
There's
there.
not many, that's one of them.
But tell me do the electronic way.
Yeah, most equine vets are lazy like me and they'd rather have pictures than.
Yeah, you don't,
draw those on.
oh yeah, drawing all the little mark.
Yeah.
Can't do that.
So I've done a few Coggins in my life as well. And you can also get a tablet, electronic tablet, computer or whatever, and sit in your truck and.
Excellent point.
And pull up the website from the USDA and down.
Yeah, so to your point, Dr. Hawkins, there are a number of companies that provide electronic certificates of veterinary inspection. We have one that is Minnesota specific. However, we lack the resources to continue to support that into the future. So Carissa and Becca are working to find us a vendor. And we will hope to continue to provide those for free to veterinarians, but there are other options, the SPS, Global Vet Link.
Yeah, VSPS, I mean, it's just the easiest way to go.
Yeah, and it's funny that you say that because I talked about this a little bit at MVMA, and one of the veterinarians said, I don't know what I did before electronic CVIs because it just saves me so much time. He said, I actually have time to eat lunch now. Right? So. Any other questions or comments?
So Courtney, considering us cervic farmers are still in business in 2020, are we gonna be on electronic?
Yeah, that'd be great. I would love that.
Okay.
Absolutely. And the advantage there, Mr. Slonick, is that for our cattle producers that use electronic CPAs, they don't have to get a permit. So hopefully we can work with server producers in the same way, just because we get that information in real time.
Any other questions?
Did you say the cost?
We don't know the cost yet,
Okay.
but we're hoping to keep the cost to veterinarians as $0.
Sure, but
That makes sense?
does it have 3 zeros or 4 or 7?
Yeah, 0.00000.
No, to the state. What
we don't-
how big of a ask do you think? Any range you can give or? you
We
just
don't
can't?
know yet, for the specific ECVI that we're looking at, we have not gotten a cost estimate. I will, as I illustrated earlier, though, we are saving a lot of money in our budget by doing this. So I'm hoping we can shift some money and still make that available to
Is
us.
it less than 18 billion?
Definitely. I don't
That's a good
think our
start.
budget is even close to 18 billion, so it's definitely less.
That's a surplus, so we'll go.
All right, thanks for the great questions, everybody.
Question, what are other states doing? I mean, I assume that we're not alone here and there's other states that are adopting some type of outside vendor.
Yeah.
And I assume we'll probably try to travel a similar direction, I would hope.
Yeah, so Becca contacted several other states, and got those figures on how many of their veterinarians are using electronic CVIs now that they've mandated that. In addition, how long did you take to roll it out? How was it received? Just to learn lessons from what they did. And a lot of those states are working with a vendor for the electronic CVIs. I believe Wisconsin is developing their own electronic version right now. So a little bit different on formatting, but ultimately they all need to be approved by our federal partners. And then hopefully paid for by the state.
Thank you, Courtney. Do we have Michelle available online?
Yes, I'm here.
Oh, go ahead.
Can you all hear me?
Yes.
Okay, sorry, you guys just got really quiet all of a sudden. Okay, so hi everyone, I am Michelle Medina. I serve as the Director of Government Relations for the Department of Ag, and also in that role, work with the Board of Animal Health. So just a quick update for you all, I won't take a ton of time. So we're just a month into session already. There's quite a lot of moving parts, a lot of bills being introduced. One thing to note is that on Monday, Commissioner Peterson was reconfirmed on the Senate floor, which is great news. We now have a confirmed commissioner, so that was very exciting, and we're glad to have him for another 4 years here at the department. There's 3 line items that got included in the governor's budget for the Board of Animal Health. So the first one is additional funding to maintain the current service level, service delivery for the board. So it's $166,000 in fiscal year 24 and $332,000 each subsequent year. So that is a very helpful one. Another one is $360,000 in fiscal year 23 and in each subsequent year for the board's emergency preparedness and response fund. As we know, high path probably isn't gone. We're, preparing for ASF and different foreign animal diseases. So this will be helpful to the board's budget and bottom line. The third one is $173,000 in fiscal year 23 and each following year to process animal import quarantines. So that is a new ask, and we were funded for that one, or we were including the governor's recommended budget. So this isn't a done deal yet. We still have to work it through the process, but it has been included in the governor's budget. So we're excited for those potential budget increases for the board. And then in this past month, the board has testified a couple of times. So the first time was Monday, January 9th in the Senate Ag Committee, and also on January 10th in the House Ag Committee. And both we gave an overview of the Board of Animal, kind of where we've been in the last year. Then just this Tuesday, February 8th in the House Ag Committee, the board gave an overview of high path avian influenza and African swine fever to the committee. And then just yesterday in the Joint Environment, Climate and Legacy and the House Committee on Environment and Natural Resource Finance and Policy, we also presented on CWD work that the Board of Animal Health is doing with the Farm Cervidae program. So that's where we've been. And then looking a little bit forward, there are a lot of bills that have been introduced. I'm not even sure how many. I don't really want to look at how many we're at, but it's quite a few. So just a few of note for the board. The first one is House File 911. There is no Senate companion for this bill, and it's to modify the Board of Animal Health. So the members that make up the board is for that one. House File 1202, also no Senate companion. This is modifying the Farm Cervidae program. So that is quite a few edits to that one. Senate File 1136, and its companion is House File 1168. That establishes the companion animal board and would take some duties away from the Board of Animal Health. And then Senate File 1411, also no House companion. This is a bill for public safety, so creating a new Office of Animal Protection for that one. And then one more is House File 1401, that was just introduced today, and that modifies the whitetail deer program. So new restrictions and edits to that program as well. There's a couple others about veterinary technicians and veterinary assistants, technology. There's 3 or 4 bills about that, or 2 bills on that, and then one about sale of pet animals in retail shops. So those are kind of the main ones we're following. That is all I have at the moment. If there's any questions.
Questions? I've got all that.
How likely, there are some things on here that are more likely to raise to the top and actually get on the floor or something. Do you have any, or is that in your role to think about that sort of thing?
Yeah, so we're not sure on that yet. I will note that today it's House File 1202 being heard today. I should know, so there's so many things around today. Only one bill is on the calendar to be being heard so far. So there's still a lot of time to session. All the bills must meet the March 10th deadlines. They must be heard in the committee before then. So it's hard. It's kind of too early to know which ones we'll get through. But there's a lot of support for a number of these bills from both parties and in both chambers.
Any other questions for Michelle?
Michelle, Scott Sloan, I'm a survey producer in the state since 1992. I have all kinds of issues with 1202. However, a real big one here is by July 1st of 2023, that bill has a clause in there that we will be demanded, ordered by statute to RT Quick our herd. I believe that causes a lot of repercussions because RT Quick is not a USDA approved herd. So that causes a lot of repercussions for other testing in this state. Has our board made these legislators aware of that? Because they probably don't know.
No, they know.
They know.
That was talked about in last year's session as well, I believe.
So if you recall, Mr. Sloan, I didn't, maybe you did, because there was so much last year too, right? But, the legislation last year actually required the veterinary diagnostic lab in Minnesota to run that test. They can't run that test because it's not validated.
Correct.
Right? They can't do that and maintain their accreditation. So they were made aware last year. And again, this year that is not a validated test.
Okay.
What I'm hoping comes to the forefront today is how that's going to even be accomplished, because I'm still not clear on how they expect farmers to collect those samples and who's going to pay for them. Because my understanding, based on Dr. Larson's presentation yesterday, was that it's a muscle biopsy. It's $90 a test. So practicality needs to come into play here at some point, I hope.
Right.
I talked to Dr. Larson yesterday. He's got his new business, pre-owned gen, I think it's called. And there's an amendment today that's going to be introduced that requires it to be a year snip. Well, that's his test. That's his skin test. And so he's an independent lab. So he has an independent lab test. It wouldn't be through university.
Yes, my point is that it's not a validated test. Somehow or another, that cannot be allowed.
Yeah, and so today the House File 1202 is being heard in the House Environment Committee. And we will have the board available for questions for that hearing.
Yeah, certainly if given the opportunity, I will remind everyone, Mr. Sloan, if that is not enough.
Other issues with this thing is double fencing. I mean, there's studies done that double fencing doesn't do anything. So why are we going to require us to do it through legislative law when we already know from other studies that it doesn't do anything? There's no security there. And I, as a herd that has not had CWD since 19, be required to possibly double fence when we have all of this positive action going on all over the state. None of these landowners are required to fence the property that a wild positive has happened on. None of them are also asked to put in their property deed that they had CWD in their farm, but yet I, as a farm, might have to. To me, all of these things got to be rebutted. Or bring a check. You can buy it. Everything in this world's got a price. I'll sell it all.
Is there any language regarding compensation for building and forcing to construct fences? And I think about other organizations or agencies deciding that they want things done differently. And perhaps MPCA wanting a year's or 15 months worth of storage for manure. So you have to figure out how you can do that. You know, just other things that can go on that once the genie's out of the bottle, how do you put it back in?
Currently, in the bill, there isn't any funding for the fence requirement.
Are there any other questions from Michelle?
Maybe just putting it out that I was just thinking, for a previous situation, there was a bill that was wanting to require a database, but because of like you can't write a bill that requires a specific database because you have to open it up for bids. Like you can't bias a bill towards so wouldn't that not be legal to do that, to require a test that it would be through an independent lab, since there isn't other options? That just
And I don't know if it makes a difference that the research in that laboratory was funded by the legislature. Is that really what, I don't know the answer to that. But I think that was.
Well, the new
was the
language
council involved.
specifies skin tests, so it'd have to be a lab that does the skin test. So that would narrow down the scope of even labs that do
So,
tests.
so they wouldn't necessarily need a lab,
But
but
by the way, Peter Larson's lab does the skin test.
it's a good point, Dr. Fox.
That's a great point.
Is Peter Larson still with the U of M, or did you say that he started his own lab?
He's done both. I was surprised that he could do both. I asked him that question.
And his,
Still funding.
I don't know, Michelle, did you hear that question?
No,
Is there funding in that bill for Peter Larson's lab?
I have not saw funding in the bill for the lab. It does say that the Board of Mental Health is required to test each white-tailed deer for chronic wasting disease using RT Quick. But it does not say which lab to use, just, it does specify the test.
I think that the last appropriated funding for that research, it hasn't run out yet, right, Michelle? Didn't they appropriate funding for the MinPro group through 2024? I could be wrong, but I think they might still have something on the books.
I would have to look on that one.
And whether we're biasing towards a particular test, if that's even the right thing to do.
Or that we're getting around the fact it's not
It's not a knowledgeable.
a validated test for which the Board of Animal Health will have to accept.
Yeah, we're
we
making
accept an
legislation on a
unvalidated test?
test that's not validated is probably. Yeah,
To Mr. Solonick's point, that we've told them, and I don't know how many different ways we can explain it, but.
I will say that Courtney will be leaving to make sure we've got our voice heard at that meeting this afternoon, and Maya will be going to another one on HPAI, so we'll have people present for all of this. But these are good points.
Yes.
I'm not real politically correct.
Maybe hearing it from you would be better then. I don't know.
Regarding the double fence issue, it'd be interesting to have comment from Dr. Scott Wells from you about his research about how prion gets introduced into these pens, and it may in fact be that a double fence and a 12-foot fence isn't going to keep a scavenger bird from flying in, and it appears that that's a significant issue. And his research may indicate that a buffer zone, a wide buffer zone outside the fence rather than a double fence may be more significant and important to preventing the contamination coming from outside the fence to inside the fence. So I think that it would be nice to, I mean, obviously his credentials should offer some more credibility rather than someone like me or Scott or any of these people making comment. But it appears as though if we're going to try and follow intelligent, logical, scientific thought, that might be an avenue to take a look at, rather than thinking we need a double fence and try and keep a bird from flying from a dead carcass into a containment structure.
Scott, could you elaborate on this buffer zone you just referred to?
His research indicates that, and he discussed this with us last week, it's not published yet, but it sounds like it will be. And Dr. Garcia, you can maybe comment if I don't quite get all his facts straight here, but it appears that in incidents of COVD developing from an unknown source, meaning that there was an introduction with an animal, purchasing an animal from a positive herd, that his research looks like, and he studied it in several different states, looks like that the prion is introduced into a negative herd by a scavenger that is scavenging a COVD positive carcass from the wild population and then gaining access to the containment pen. what he said last week was it's likely that up to 70% of new infections may be caused by that. And so I think that we're getting ahead of ourselves thinking that a double fence is our answer, which it likely is not, and it puts an extreme expense on these deer farmers. And it probably does nothing or very little. It's probably not something that is going to create a significant difference in what we see in conservative farms. And that possibly management issues inside the pen plus a buffer zone, so the scavengers aren't as likely to want to enter that pen over the top of the fence might be a better answer than spending much money on double fences, which probably isn't going to keep a crow from flying into a containment structure.
He talked about wooded areas versus cropland versus just open fields, that type of thing.
So scavengers less likely to want to go in there to drink water or grab some feed or look at the deer.
But, and not to put Alex on the spot, but as our tribal representative, I just wanted to say that I think it's very evident that this is a request coming from our tribal partners for the double fencing. So
they should look at that research and talk to Scott about that, because that was very interesting. And you want to do things that make a difference. You don't want to just spend money to spend money to spend money. You want to do things that make a difference. And there are many things that can be done that don't cost the taxpayers any money and will be way more effective.
Your partners are requesting the double fence.
yeah, Chairman Dupuy with the Fond du Lac Tribe gave a testimony yesterday that that was a request from our tribal partners.
So if I understand you, you're saying they want us to double fence, yet they want to bring untested elk in.
You'd have to take that up with them, but
I got that right.
Just if I may, just really quickly, that maybe this is a Jessica Fox just side note, I guess. So one of the things I talk to vet students about and other veterinarians is, when it comes to legislative stuff, my opinion is if you're not at the table, you're on the menu. And, you know, one of the things that I heard about the sessions yesterday was that folks were hearing emotion. And I hear you, Dr. Josephson, and as veterinarians and as scientists and sometimes as producers, we're like, but these are the facts, but these are the facts. But these aren't experts. They're people that we brought up from the population, and they hear stories and they hear people's hearts. So having producers and having representatives say, I've been farming since 1992. This is my life. My kids are on this farm. I bring my little girl into the turkey farm in her little shorts. It is completely adorable. Like those things are what touch people's hearts and have you listen, because they hear the emotion of the other people. So if you have stories, I would just encourage you to not depend on other people or state agencies to speak for you, to speak for yourself. Listen to my voice. That's my gun. That's good.
And I think that a number of people in this room plan on heading over there today in the hopes that they'll be able to advocate for themselves, right? Thank you, Dr. Fox.
Yeah, I thought that's a good point. I think we do tend to focus on science, but you're right. You gotta make them care. Yeah.
With that being said, our survey producers are up next. Any last questions or comments for Michelle?
I have a question. I'm Paul Anderson, and I wanted to comment on Senate file. It was your Senate file 1411, which is the companion animal protection bill. And I just wanted to make sure this group is aware that that particular bill was written by two police officers in the Minneapolis Police Department, Michael Fry and Jenna Washi. And
Is that the OAP?
that's the OAP.
Right,
Did I
you
say
said,
the
you
right thing?
said CAB.
I'm sorry,
Okay.
Office of Animal Protection. I'll start back where I should have. So it's on the Office of Animal Protection, and I wanted you to know that the MPMA, the Minnesota Veterinary Medical Association, and the Small Animal Welfare Committee of that association have both reviewed this bill and stand behind it as written. We really think that this would fulfill a niche for animal welfare and animal health in the state, and that it would complement the Board of Animal Health and not take anything away from the Board of Animal Health. And so this bill as written, and I see it sponsored by Senator Champion. Small animal welfare, but the board for MVMA. And we think that this would be a good bill to support. It was also discussed at MVMA Governmental Affairs Committee, and that committee officially supports the bill as well. This is the first time that we've seen an actual number on the bill. So that's good to see. And I just wanted you to know that we think this would be a good thing to support. And we don't think that in any way it would affect the Board of Animal Health, except in a positive way.
Thank you.
You bet.
Thanks, Michelle. We appreciate your time today.
Thanks.
Whoever wants to go first.
Yeah.
All right, greetings, everybody. It's kind of tough to go after the heels of that, but I'll give it a shot. So we were asked to give a presentation on the day in the life of an elk farmer. But really, it's kind of a year in the life. Every season is different for us. Every season ramps up with the work that we have to do. I'm an avid picture taker, so of our animals, most of the animals you see in these photos will be from our farm. But anyway. So I'm just going to start real quickly with why we raise elk. It's not only about breeding stock and meat, but the antlers add a whole new dimension to what we do. And the reason I bring that up is because it also impacts what we do during each season. Midst of that now, this is our slowest season. So we are, elk are on a maintenance diet right now. On our farm, we pull all the grain actually December 1st, except for our calves that are rapidly growing. In nature, their metabolism slows down, and so we treat them the same way in the farm. We feed mostly hay. We feed hay year round, but of course that becomes even more important in the winter. And we even lower the quality of the hay because they would get too fat on the higher quality hay they're getting when they're producing. But we do offer a loose mineral supplement to make sure that they're getting all the vitamins that they need. Paperwork and testing. We just got done with our TB accreditation and brucellosis certification in December. It's a good time for that. The cows are in their just after the first trimester of pregnancy. There's no antlers on their heads, so it's a good time to get that done. We do our deworming at that time, typically when we have them in the shed. Do our herd inventories. People can do that year round, but for us, it's easiest to do when we have them in the shed and we can get the verified inventories with our veterinarian. We're securing trophy bull contracts for the fall. We're talking to the contractors out there to get that all lined up. But political activities have increasingly taken over much of our time from January to May, and actually it's kind of a year-round thing now, unfortunately. But as Scott was talking about starting in 92, we started in 93, so we've seen a big evolution of things. When we got in 92, it was all about brucellosis and a national program for that. And so we started out buying animals that were from brucellosis, or excuse me, TB-free herds, even back then. And that issue kind of went away, but of course CWD has not gone away. This is a time that we do pregnancy testing. Again, they're right after their first trimester. Moving cows after the first trimester. If you're selling breeding stock cows, bred cows, this is a good time to do it. Some people also move bulls at this time of year without antler. It's a little bit easier to move them. We don't on our farm, but some people do. And if you're selling calves to someone, this is a good time. They're past weaning, so they're well on their way to regular food, and it's a good time to do that. And this is also our first opportunity to see some antler on the heads of the males. It's actually the spikers, the year-old elk that start growing antlers first. And you can kind of see the guy on the right-hand side, you know, they just kind of start pushing up, making a pedicle. The guy on the left-hand, excuse me, other side, on the right here, 265, he's up a little bit farther. But they just kind of keep creeping up as time goes on here in the winter months. You see March and April, that antler, it's another spike antler guy, he's kind of stretching out a little bit. We started increasing the feed. I know we on our farm started increasing the hay quality on February 1st. We're gearing up for the antlers to start growing again in March. We're starting to introduce grain again about the end of February, early March, because the cows now are going to enter their third trimester. So we're busier going on into more pens and feeding, preparing for production season. At the end of April, we vaccinate our cows because our calves are coming in 3 to 6 weeks, and we want that protection to kind of filter on into them as they're going through the summer months. This is also the time that we record what we call button drop. You can kind of see the guy over here, he's got kind of hard pieces of antler on his head. He hasn't dropped his antler yet, where this guy probably dropped about 2 weeks ago. And so you can kind of see how it starts coming out of the head. It's kind of a big mushroom that comes out of the head. But we like to record when those drop because we count days. If we're velveting the bull later in June, we kind of like to count the days to kind of see if they're kind of in that prime range for the velvet market. But it's also the time where we decide which bulls we're going to actually grow out and which we're going to velvet. Oftentimes we'll kind of sort them. The hard antler bulls will stay together and the velvet bulls will stay together, so there's no damage to that velvet as it's growing. So May and June, everything it really starts to ramp up because hay is such a big part of their diet. You also have we're also trying to secure hay, maybe bale our own hay. Calves start coming. You can see the antler is really pushing out. Calving starts mid-May through July. Of course, that's a very busy, 6 weeks for us. We try to wait, at least on our farm, until the last possible minute to move them out in their summer pens where the grass is about as tall as it's going to get before calving starts. So about mid-May, we do that. Usually there's a calf or two shortly thereafter. We're tagging calves each night. We come, they typically calve during the day. It's very unusual for an elk to calve at night, overnight. So you want to watch them though. If you have a cow that's still really struggling and it's 10 o'clock at night, she's probably got a problem. You probably have to bring her in. So there's a lot of work with that. On the top of that, we're velveting bulls at the same time. We're getting out to that 60, 70 day range where if we're going to velvet the bulls, this is the time to do it. So on top of going out and tagging calves at night, you're also bringing bulls in at night to velvet because the days are so hot and it's quite stressful, to do the velveting procedure, and you want to minimize all possible ways of stress. So we do it in the cool of the evening so that they're recovering in the cool of the evening as well. So the handling facility, that was a question that someone had. So you can see there's a series of cubicles. You can see this gate would push them forward. This gate will sort them. So we'll fit maybe 2 or 3 animals in a cubicle. You can see the guy over here, he's in one of those cubicles. He's just waiting his turn to be pushed forward. We would never go in this cubicle with them. You can see how everything sweeps the next guy forward. Oops. So squeeze chute. So ultimately your goal in the handling facility is to get up to the squeeze chute. So this is a hydraulic, sorry about that. Hydraulic chute. So the animal comes in there. You can see the guy at the bottom of the page here. He's in the chute. You can see how his head is above the huggers. This actually hugs them up and lifts them up off the ground. So they're completely immobilized there. We do put a halter on our bulls when we're cutting antler, just, you know, so that we don't have the sudden head jerks and that kind of thing. And so this is a guy that was brought in for velveting. He's all ready to go. So removing velvet, and I don't have time to get into that, but you can see we bring in usually a group of bulls to do at one time. We have to hang the antlers inverted because the blood will run out. We do put them immediately in the freezer when we're done, and then we hold on to wait till they're all collected, and then we will sell them later in the summer. Another picture at the bottom of the page of an animal that was brought in for velveting. So May, June, these are the pictures on the right-hand side are pictures, again, from my farm. Those are out my east window and out my south window. I wake up in the morning and I look out and I grab my camera in my pajamas. I go out in my back and I start taking pictures. But it's an amazing way to wake up in the morning. July, August, again, ramping up. You can see this guy by early August, they're scraping that velvet off. You see kind of strips kind of hanging down from him. It's just about completely rubbed off. This is a time where we velveted the bulls. We're watching our animals for signs of antler damage or maggots. So you can see this guy here, he was cut earlier in the season, but they get some regrowth that comes back. And there's lots of nooks and crannies for maggots to get in there and cause havoc. So we really, every day we're monitoring for that. And that guy over here, you can see he's kind of weepy down there. So we're watching for that sign, because that means that maggots probably got underneath that burr. You want to get that treated right away. So every day we're watching for that. So also finalizing that paperwork, the CVIs for moving those bulls, because as they're hardening off, we're going to be shipping off bulls for the trophy bull sales. Also breeding stock. So this is a guy down here we grew out last summer, like, man, would he make a nice breeding bull for somebody. And so this is the time, July, guys are coming over. If they're looking for a breeding bull, they're looking for that kind of bull to buy from you. This is also the time that they come and usually pick up the harvested velvet. It's usually all been collected by that time. And so the buyers come in typically August to pick that up. Also a time for cutting antler, scoring antler. As you can see, it's a family fair at our house. This is not only cut antler, but we're measuring it. We're adding up the score. It's also the time for the International Antler Competition, end of July. My husband and I were fortunate enough, we won a couple of the classes last year. This was our mature, typical picture, the bull we call Vision. Also the time that we're hauling trophy bulls, end of August, last week of August. It's not uncommon for my husband to be making 4 trips to Colorado in that 2-week span of time. This is Monarch Pass, I think. Our son is outside the truck there with that trailer. This is a picture then of the bull exiting that trailer into the preserve. And then we enter the rut, again, a busier time. So especially, this is a good picture of explaining why we cut that antler off. The family affair picture I was talking about. You can see they like to spar. Their emotions are running pretty high in the rut. And we don't want them to injure each other or us or damage fence. So you can see the picture, this picture, you know, they're just playing around. They're not going to hurt each other or hurt us in the process. In breeding season, so Labor Day weekend is on our farm, the big weekend, where we pull, we're gonna wean those calves. They've been on their mother since May, June. We wean the calves, we place the cows into their breeding pens. As we're weaning, we insert seeders for AI breeding, if we're gonna AI breed that year. You draw semen at that time of year. Fun fact, elk are not fertile when they're growing antler. So right before breeding or right after breeding is the time that we pull semen if we're going to do that. And breeding season usually runs from about Labor Day weekend till mid-November or so. This is a time where the phone starts ringing for meat sales. As I was showing you earlier on, meat is a big market for us. I think people think about hunting deer and they think about eating elk. I don't know, but what it is. But anyway, we usually pull off a group of cows that we're going to process, you know, when we're sorting animals for breeding. So they're in their own pen, and that's when we kind of finalize the halves and quarters, match them up with our customers. And it's mostly all cows. The bull would probably have to have antler that is really, really bad for you, not it's to butcher him, or it's damaged beyond repair. It's mostly all cows. Poor breeders, poor temperament, genotyping. You know, we've gotten into the last 10 years where if it's between 1 or 2 cows, we're going to look at her genotype, and the one that carries the more resistant genes, she's going to stay. And it's a seasonal thing because we don't want to sell meat during, you know, the time that antlers are growing, or we don't want to process animals. We don't want to process animals when they're nursing their calves in the summer because they're busy feeding their babies. And we don't typically like to send them to slaughter when they're, you know, the second, third trimester. So for us, it's kind of a seasonal thing in September through December. And then mid-November, we remove the breeding bulls. And again, we sort our animals and kind of bring them up closer to home. So there's just a lot of area to keep snow removed and gates open, especially in a winter like this. So we kind of tuck everybody closer to home in those winter months. And then again, kind of starting over, we remove that grain December 1st. And this is actually a group of that year's calves. So you can see how much they grew in just that 6-month time. They're just little miniature elk. So thank you for taking this journey with me about why we raise elk and what we do and what our life is like in the elk farm. And I would entertain any questions.
Brenda, when you cull and those cows that you're marketing meat, that's a big part of your CWD surveillance is done with those.
Yeah, exactly right. Yeah. You know, people talk about CWD and think elk and deer are pretty sickly. They're not at all. There's very little animal loss on our herd. Very little. It's mostly all done through slaughter surveillance. Brittany?
Great presentation. Thank you so much.
Thank you.
The velvet market, what's that look like now? So I've read a lot of research that's ongoing with velvet in reindeer and elk. So what's your market look like right now? Who are you selling the velvet to?
Scott Salonik. He left. We're selling it to people like Scott. There's another Brian Wagner, you know. They actually go around the United States buying antler. Okay. And so they actually just come right up to our place, our freezer, load up, hand us a check and go off. So it's a very simple thing for us to sell. And then they're kind of intermediary between the buyer and us.
And they're using it to grind for supplements or something? Mm-hmm. Yeah. Mm-hmm. Then you have the hard antler too that is torn up dog chew toy.
Yeah, we didn't talk about that, but yeah, you know, the antler that you saw that was in the grass, yeah, that does typically go for dog chews. It's a big thing.
How do you take, just one more question, how do you take your calves? I mean, are you putting the cows in a pen or how do you safely do that?
You don't. We've actually gone to it. It's just a lot easier, a lot less stress for us and the animals is we end up tagging when we wean. So we're still meeting requirements, you know, by December 31st. Well, we're doing it Labor Day weekend. So we're well within our window. Sure. And then we do DNA testing. So we know exactly who came from who. Sure.
It's life when you do that weaning, but what about if you were to do that as a newborn? If you were forced to have to do that as a newborn, you put that big tag in a newborn calf, the newborn, the ear is soft, they're very tender.
Yeah, they drew.
Shred their ear and
yeah. You can put you know, the news tag, they make that nice little plastic 1.5-inch tag. That works really well. You can't read it very well, but those are a lot easier on the ear. So those are easy to do. It's just, getting in there to Erica's question, yeah, you gotta be fast. Because, God help you if the very aggressive mama cow is the first one to calve, because she'll be after you the whole season, no matter who you're trying to tag. So, it's just, they used to meet us at the gate ready to kill us when we went in to feed them, because they knew we were gonna come and disturb everything. And when we went to just tagging when we weaned them, they're calm, they're not getting stirred up in the heat of the day. And everybody is, that's a big thing for us, is really keeping the stress low. And I know it is for the deer breeders too. It's important. It really helps.
Sorry if I missed this, but how much does a calf weigh when they're born?
About 35, 40 pounds.
Thank you, Brenda.
Thank you.
Thanks. Good morning, everyone. My name is Fier. I'm the president of the Minnesota Deer Farmers Association. I live in Southwest Minnesota, actually near Dr. Capeen Fox and Dr. Josephson. Actually, my sister, Dr. Jenna Fier Dirks, also works with the board, and my dad, Mike Fier. So some familiar faces anyway. I started raising deer in 2001, which puts me at about 22 years of raising whitetail. I absolutely love it. In addition to that, I also raise, I've got 1,100 head cattle feedlot, and then also about 150 whitetail deer. I raise 800 acres of corn. I have two daughters, ages 10 and 11. Like I said, I've been raising deer for 22 years. It's gotten to be kind of a family affair. My kids absolutely love it. We get a lot of traffic. We get a lot of drive-bys. People drive by and look at our deer. We get I talk at a lot of schools, although I'm not very good at speaking in public, but I get a lot of schools will come out to the farm. I've had nursing homes, Boy Scouts, all different kinds of people come out to the farm, and a lot of times we'll take them out to the pens and we'll go grab a little baby fawn or whatever, and we take pictures with it, or we feed the deer a treat. There's always we always had one doe that I could have a marshmallow, and I'd hold it up in the air, and the doe would stand up on her feet, and then I'd pretend like she's giving me a hug. And the kids, I don't know, the kids and the families just love that. But as far as a typical day in the deer farmer, that's rather simple, I guess, when you think about it. I guess, obviously we feed them, check feed, water, herd health, maintain our fences, interfeeders and waters, do herd counts. But it's obviously much more complex than that. And I'll kind of go back to kind of like Brenda did, kind of start like in January. January, February, March, a lot of times I do, I'll do like a feed sample, make sure my feed's up to, where I want it to be nutrition wise. And nutritionally, it's compared, closely to like a small calf ration, like a 300-400 pound calf. It's similar to what we feed, it's just different ingredients. And then also a good quality hay, or browse, is always good. And then I'm always looking into, ordering my tags for my newborn fawns. I tag my fawns with, like the year, 23, like, so this year my tags would be 2301 on up. And then I change colors every year just so I can keep them identified. By color code too, I can tell their age. Looking into, my vaccination protocol. If I need to change anything there, I don't do a lot. To protocol, with myself raising cattle, near where I raise my deer, I do a couple different vaccinations, I guess, to. One other thing I also do is I put like ag lime in all my deer pens. I spread, especially by the feeders and waters and stuff, where the lime helps neutralize the soil. the bacteria. What else? And then, yeah, starting in about, May 15th, May 20th or so, we start having our fawns, start being born. Fawns typically weigh between 5 and 10 pounds. And I don't have the trouble that Brenda maybe does with, our does are, they're fairly calm. The does that would chase you around, if they didn't like you messing with their baby, they went down the road. So yeah, it's, so at that time, after the fawns are born, I generally wait at least 24 hours, so the baby can get, their first round of colostrum from their mother, if not 2-3 rounds of colostrum from their mother, before I touch them. Then we tag them, I spray iodine on their navels, and their feet. Give them a couple of vitamins, a couple different things of vitamins, and then if I ever have, if I have triplets, or say a fawn isn't nursing, or maybe the mother didn't take care of it, in those cases, I always pull those fawns off and bottle feed them. And I actually have a lady that helps me bottle feed, because that's like having a baby child around. I mean, you gotta have, a lot of people feed like every 2-3 hours around the clock. Which maybe isn't necessary, but is probably best for the fawn. Yeah, so after fawns are born, then as fawns are being born, I do every 2-3 hours, I'm out of my pens, walking around, checking for newborns. So I can identify each fawn. It's important to me to identify each fawn when they're born to the actual mother. So I don't have to DNA everything necessarily. So I can save that cost. But sometimes they do actually do like a pregnancy test after we breed them to see which ones are pregnant. And I guess part of that reason is that time of year I'm busy in the field, like with corn, spraying my corn or planting my corn or whatnot. So helps me where I don't have to be in the pen 24/7, I guess. So sorry if I'm bouncing all over the place here, but so yeah, as the fawns are being born, our bucks are starting to grow their antlers, which in a deer farming world, that's the funnest time of year. We're having our babies on the ground, antlers are growing, we're seeing our fruits of our labor in a sense, where, all these bucks, or the way we breed our deer, obviously the current years should be your best genetics on your farm. So we always hope that our antlers are a little bit bigger, a little bit more antler characteristics, how we want them. So then we get into, that's kind of busy time too, like June, July, and August. Even into September is the busiest time of year where I get a lot of traffic, a lot of people coming out, a lot of the tours. I live on an intersection on a gravel road, so it should be tarred because there's a lot of people that drive by, and I really like the feedback they get from other people in the community and stuff too, when they, I've had numerous times even where you'll be at an event somewhere and it's like, well, I live by Minneota, Minnesota, and it's like, I don't think I know you. Do you happen to know the guy that raises deer there? Well, I guess that's me. So whether that's good or bad, I guess, but it's for the most part, it's been very enjoyable. And in the summer here too, it's a lot of times my kids and I, we go out to the deer pens and we sit on a 5-gallon bucket with a bag of marshmallows or some treats or something, and it's very relaxing and calming, I guess. Just to sit out and interact with the deer and seeing them mingle around and interact with each other. I really enjoy that. And I do have a ton of pictures, but I don't have a presentation like Brenda did, so I apologize, but I just have a ton of pictures of, my kids interacting with the fawns and their friends interacting with the fawns, and those, people that come out to my place, families and whatnot. So I got a lot of memories, so that's pretty neat. But then we get into like September, I usually by now, I guess I should back up a little bit. I've been marketing my bucks as they've been growing, say like around July 1st, the bucks, you can kind of see where they're going to finish up with their antler characteristics, where they're going, and you got a good idea where they're going to finish out. So I started marketing the animals to previous customers or previous buyers, sending pictures, negotiating price, time, whatever they try to accommodate them as well, which is kind of fun too. Then we get into, September, in that area. I try to wean my fawns around September 1st. I usually pull my fawns from their moms, and put their official identification in, maybe give them a round of a vaccine. Then I actually put the fawns back in with their mothers for a couple more weeks because this is a very stressful period for them. And deer do die. I mean, so we try to, like Brenda alluded to, we try to be as little stress as possible. And I feel like in doing that, putting the fawns back in with their moms, helps relieve some of that stress, and then another week or two later, then I'll separate my fawns from the mothers for good, put them into their, I keep them all the same age. I try to keep them all in the same pen. And then continue to, market bucks. Then I try to sell my bucks by the end of September. So hopefully I have a buyer by then, get paperwork going. And then I was gonna point out, like with my bucks too, I try to get into, only work with people that have, I guess what I feel is, I don't know which way a big area. It's actually hunting. I know some people get criticized because they're too small to preserve, or, maybe it's not ethical. Some people say we hear in the public. I always try to market my animals to, 1,000-acre preserve of woods and river bottoms. And, I've had a deer in a state for, I think it was 4 or 5 years, and nobody ever seen it at this preserve. Not on trail cam or nothing. And it had a microchip in it, and they found it, looked like it got gored by another buck, and it died. So it's, to me, that's a real experience there. If you can't see a buck for 5 years or ever see a deer in these woods, I guess that's what I want to work with. And then, I've been working closely with a local business, in Marshall, actually. It's called Action Track Chair. It's like a wheelchair with tracks, basically, which very good group of people. I'm trying to get lined up with them where, they donate a track chair to somebody in need, of course, and trying to incorporate something where I could donate a deer to that same person. Because that's my ultimate goal, is to be able to donate a deer or 2 or 3, whatever it is, whatever I can afford, to somebody that, make a wish, or wounded warrior. It'd mean the world to me to be able to, 15-year-old girl dying of cancer, terminal cancer. She's got 30 days to live, and her last dream in her life, her goal in her life, was to harvest a white-tailed deer. That's where I want to be, and I'll get there. As long as we can stay in business long enough, of course, but, that's where I'm headed. And I know many other of the breeders are the same way. I mean, we're passionate. We love animals. We love taking care of animals. So that's the side of hunting, I guess, that I wanted to talk about. Then I'm also, throughout the year, I'm also talking with other deer farmers, whether it's in the state or other states, about what I'm going to do for breeding season. Breeding season starts about November 1st. Actually, Dr. Scott Josephson does my breeding. I try to pick out deer that have the antler characteristics that I like. Good maternal lines. I guess some studies have shown that the maternal side of a deer is more important than the father side, as far as, taking the genetics onto the next year. So try to focus on that. And now it's gotten into where the genetic resistance has been huge. So now I try to take the best genetic marker deer with the best antler characteristics that I desire, and breed that into my herd. So it's kind of a fun time of year as well. So that starts about November 1st. You know, I breed about November 10th, you know, generally. And then we do the laparoscopic AI with Dr. Josephson. Then about 17 days later, I introduce a cleanup buck or a backup buck into the doe pens to clean up or to breed any deer that didn't conceive to laparoscopic AI. And then, well, if I don't prig test them, you can still tell who is the sire to your fawns by the date that they were born, whether they're laparoscopic AI'd or live bred. And the average gestation for a doe is around 196 days. So if you breed about November 10th or so, that puts you into like May 20th or so. So that kind of brings it back full circle there. Sorry if I bounced around there a little bit, but I think I covered most of the stuff I wanted to talk about. Also wanted to mention too, just how essential we are to our communities. You know, and how I kind of got started in this deer farming business was we had an old grove, you know, that wasn't in agriculture production. It wasn't going to be anything for our cattle operation. So I was taking marginal land and try to utilize it and, you know, try to generate a revenue stream. And that's kind of where it all started. And then it kind of, you know, went from there. But I feel we're so important to our local communities with, you know, all our vet supplies, our feed, our fencing, or, you know, it's so critical to have the small farmer stay alive, I believe. There's big commercialized dairies and hog facilities, and nothing against them at all. It's just the little guy's just as important as the big guy, in my opinion. You know, we have an impact as well. But I just wanted to highlight too, you know, I love deer farming. I love every aspect of it. But it's gotten to the last few years have taken a lot of the fun out of it with all the regulations, and you know, I feel like we're currently just we fight every day to hopefully be alive tomorrow. You know, because I literally don't know if I'm going to be deer farming tomorrow. You know, and it's hard to do, you know, make any capital expenditures, fixing fence, you know, fixing your waters or feeders or whatnot, because you know we've got a lot of time and money invested into this. You know, over 22 years. I was just adding up the other day. I mean, I think I got well over $1 million into this deer herd if you consider feed, vet, you know, everything. You add up all the expenses throughout the 22 years, so it'd be very devastating to have to put up double fence or, you know, add spend a lot more money on our property, I guess, is what I'm getting at. And, you know, and we feel we're the solution, not the problem, you know, and I strongly believe in that. And we're willing to work with people. We want to be part of it. And I truly think we can solve CWD. So, with that, I think that's about all I have, but I'd be happy to answer any questions you may have.
Your boss.
Thanks. First, I just want to say thank you for giving us an overview to both you and Brenda. Dean and I have both heard from Brenda and then another deer producer, but the rest of the board members haven't had the opportunity to hear just how you guys make a living. So, and my, you know, growing up, I'm the 6th generation on my family. Even, you know, just talking about and sharing what you do, that's so important. And even for your local community, you know, your neighbor might know that you're a deer farmer, but they don't know are really an advocate for yourself as a farmer. So my question for you is, and it's for you and Brenda, or both cervid producers in the turkey industry, and you know, being a corn producer, you would know this, but we have checkoff dollars. And those checkoff dollars go for research and promotion, which is really important in agriculture and for our industries. And I don't believe that you guys have anything like that. Is that correct?
No.
Yeah, so, like the research that Dr. Nicholas Haley is doing, could you maybe just briefly just like elaborate on how important that is for your industry? Because you guys don't have the checkoff dollars that the rest of agriculture really does have. I think that's important for the public to understand as well.
Yeah, I agree. Research by like Dr. Haley and Dr. Seabear, I think's our lifeline. I think the way legislation and everything's going, I think that's our, one of the main avenues we have to go down, because we truly feel we can beat this. Or eliminate it or eradicate it to the point like they did in the scabies. So in my opinion, it's so important, and, hopefully they can get enough funding to continue to keep doing their research. Because I think it's vital to our existence.
Yeah.
And participation and taking advantage of that funding, and I have lots of conversations with Scott about this, by our farmers is really. I want to give us more funding for not using it, for one. Two, maximum participation is going to give us maximum data, and it's also going to help build Dr. Seabury's database, right? So that we get more accurate information. So Addie and I are going to carve out some time to individually call farmers and ask them to participate in
the research study. Yeah.
I'd also like to add that what you're doing and the research and everything and participating in all that and understanding some very difficult things to understand about genetics.
Yeah.
You guys know more than a lot of people right now.
Yeah.
But you are the hope for the wild deer. I mean, we
I
really
agree.
need to bring that up. You're the hope for the wild deer because this thing is in the wild and it's not going to stop. And so what we can learn on the farms is the hope for the future of hunters.
I agree. I think that's critical. I mean, I think, and we want to be involved in that because there's nobody in the world that wants to solve CWD more than a deer farmer. So we're trying to do what we can. We're trying to do a research. We've worked with the Board of Animal Health in some cooperative agreement money to do the genetic resistance testing. And also want to thank the Board of Animal Health. You guys have been amazing. You truly have. You answer all our questions and you've been great to work with. And hopefully we have a voice at the Capitol, and so I just want to thank you for that.
Scott, I've got a comment too to follow up with Dr. Hawkins. I think to this point, I think we try to put the wild deer and the captive raised deer in the same group, and they're not. You are a turkey farmer. Turkeys were bronze originally. The domestic turkeys that you raise are the same bird, just bred up from the wild bird. And I think that, and the industry, depending with the natural resources, the Board of Animal Health, the ag production industries, look at those as two different entities. The farm-raised turkey that we all enjoy eating and the wild turkey that the hunters enjoy hunting are seen as two different entities. I think as a public image and as an approach that the legislature would need to take a look at this industry is, we need to completely separate the thought of the captive raised deer from the wild deer, because as we improve the genetics in the captive deer, just as the turkey genetics were improved to create the bird that we now consume today as food, they're becoming two different groups of animals. And so we can look at the captive raised deer, and we understand in this room and as a scientific community, looking at the scrapie model, we can take care of CWD within the captive raised deer, but we need to look at that separately than the wild deer. And that leads to the next discussion of what's going to happen with who's going to have oversight over the captive raised deer. And that should be the Board of Animal Health, just like the Board of Animal Health has oversight over the captive turkey industry. And the DNR has jurisdiction over the wild turkey industry, even though they're the same animal, they're susceptible to the same diseases. Bird flu affects both of them. And I think that, and again, I'm a little bit of soapbox here, and I apologize for taking that time, but I think that part of the problem down in St. Paul is that we lump it together. And we need to find some way to convince the industry outside of our world that they need to be seen as separate entities, just like the turkey industry is.
Yeah, we are considered livestock, but yeah, we get pushed to the side and, kind of ran all over.
So we have two models. We have a scraping model for CWD and we have the turkey industry for how we need to look at those two animals that are same species but different purpose animals.
Correct. Thank you, Scott.
Do you sell breeding stock with a 5-year trace back, of something that you sell or that you're responsible for, the way the law is now? Do you still sell breeding stock, take that risk?
Good question. No, I do not, just for that reason. It's definitely, it's been a huge financial burden for me to not be able to sell breeding stock because that was one of my go-to, to be able to sell bred does and, we're CWD certified. I've been in the CWD program since 2003, I think, since it first started. So no, I haven't sold any breeding stock for, I think it's 3 or 4 years now. Just for that reason of the trace back. Mark?
I'd just like to add a comment about the checkups. One of the things that our association, we spend a fair amount of money on is a lobbyist down at the Capitol to keep us right. Hey, this bill's coming out, that bill's coming out. We're farmers. We don't sit on top of legislation. We don't keep up with that stuff. If we didn't have to pay him so much money, researchers like Dr. Haley, Dr. Seabury, Dr. Bastion, all the different research, we could be donating that money towards those causes. But we can't because we have to have that lobbyist to keep ourselves alive. We're fighting to survive. And that's where money's gotta go first. After that, if we didn't have that worry, quite a bit of money we could donate to those other causes.
Thank you.
It almost seems like it's not about CWD, in a way, it's almost like it's a political or financial agenda, and that's the frustrating part. I started a legal, viable business in 2001, and now I'm getting, regulated out of business. It's a regulatory taking of my private property rights. And that's the frustrating part, is you put common sense and principle into this, and that's why I'm standing here today. That's what I believe on, is the common sense and principle factors of this, that if we use science and follow the science, we have deer farms who can get hands-on with their deer, numerous times of the year if we need to. We're willing to do the work. We're willing to help out, because like I said, we want to find a cure or, eliminate or suppress CWD as far as we can, to maintain, business. So any other questions?
Are there any from the back?
I'll just throw one comment out here real quick, just so, and on a little bit lighter note, I'm a deer farmer. I also am a benefit advisor. Been farming for, I'm not gonna disclose my age, a very long time. For those babies that are protected by their mom, I have a 4x4. I drive up to them and throw them in my 4x4, and then the mother runs around it. That's how I take care of them. Because she happens to be a really good producer, and I'm not gonna take her off my farm.
Good idea. See, age, you'll get wiser with age, right, Deb?
So when you cull your does, I would assume that's where you get most of your CWD plus anything that you lose.
Yeah.
What do you do with the meat from that? Do you have people like Brenda does where you market it then?
No, I usually don't market I guess I've never marketed my own meat, but I have taken meat to, it has to go to a USDA inspected plant, which there's one in Foley, I know, and there's one in Wisconsin somewhere, but and generally then there's a private producer, whatever, buys the meat, and it's all CWD inspected and whatnot. So yeah, good question.
Like on my farm,
everything that I call out goes right back to my family. I've got 7 kids, you know, how much beat 7 kids. I mean, you know, I mean. Better. Yeah.
So in closing, you know, if anybody, I mean, we're here to help. We want to work together. If anybody needs anything or wants anything, or if there's anything we can do to help, please reach out, to myself or any other board member. And with that, thank you for your time, and, we'll go on to the next speaker.
Thank you, Scott.
Thank you.
So we're right before lunch, but do we have Albert to give us a quick update on the diagnostic lab?
Yes, here I am. Can you hear me?
Sorry, Albert, you're right before lunch.
All right. So I don't have a PowerPoint, but I'll share some updates from the lab. I'm sure everybody's a little tired by now and very hungry, so I'll try to be brief. Again, thank you for the opportunity to share some updates from the diagnostic laboratory. I'm going to talk very briefly about personnel, testing activity, equipment, and projects. So hopefully I'll just go through all of those quickly. In terms of personnel, we talked last time about how we were almost ready to open the VDL director position. Finally, that got open last month. So that is now open, and we're looking for candidates for the director. So that's great news. The search committee put together a flyer that was circulated internally today. And so I'm going to share that with Erin and ask her to circulate it through this group. So you can all see the flyer and share it with others. And there is a link that you can actually go and see the position description and so on. And please help me in trying to recruit as many good candidates as possible for the director position. So that's the update for the director position. We have another important position open. Again, last time we talked, I believe it wasn't quite open yet. It has been open, this one, for a little longer. That's the food animal diagnostician position. And that one, like I said, it's been open. We have a couple of applications, and we haven't done interviews yet, but it's encouraging because I know those positions are hard to fill. There are other laboratories having also having problems filling this type of position for a food animal diagnostician, and having several applications already in, it's encouraging. So hopefully the next time that we meet in the next quarterly meeting, I can tell you about us doing interviews. I don't know if we'll have a hire, but at least us doing interviews and getting much closer to filling that one. And finally, again, we talked a little bit about staffing issues at all levels, also with our staff at the different laboratory sections. And I'm happy to report that in the last couple of months, we've been able to hire several people in different sections. So that's been good. And we've only lost one person who has left for a different position. So we're doing good in that regard. I don't know, I need to investigate a little bit more in the people that got hired, on how they learn about us and so on, because we made some changes about how we advertise some of these positions, and I'm not sure we can claim that those changes got these people to apply. But it'll be interesting to see if that's what happened, or like before, people ended up learning about us from other employers and current employees, and so on. So, but anyway, the good news is we've hired people in several different sections, and we are a little bit better staffed now. Now we're in the process of training these people, but better than the last time that we met. So that was for personnel. In terms of the testing activity at the VDL, I just looked at the last 6 months of fiscal year, first 6 months of fiscal year 23, or last 6 months of 2022, to look at our testing volumes. Our feeling is that we're really busy. but like I said, we were understaffed in some sections. But really, the numbers also show that we're really busy. We've tested 9%. We've run 9% more tests in the past 6 months, or in the last 6 months of the year, compared to what we did in the same months the year before. And that's something that seems to be stable. Every single month, we've been running more tests than we did in 2021. 2021 was not a good year, but 2022 has shown to be consistently better than 2021. So that's good. In general, we're up in most of our sections, so more PCR tests, more bacteriology, more serology and virology. MPTL, the Wilmar lab is a bit different because they are very busy with HPAI avian influenza tests. Obviously, in the past year they were, and not so busy in the more regular serology and bacteriology tests. But that is to be expected during an outbreak. And then perhaps the one that I have to take a look at, the one that perhaps is not increasing as much, is our lab for testing milk, the other health lab. I need to take a look at that and see which tests are not increasing for that particular lab, because it doesn't seem to be going up as much. A couple other things. We have increased the frequency of bovine PCR tests. This is something that we had to cut down. We had to cut down from 2 times a week to 1 time a week during the pandemic. We're back to 2 times a week now, trying to help bovine vets a little bit more with offering those tests 2 times a week. And before the meeting, I asked for some input from our veterinarians, our diagnosticians, on what is it that they are seeing, if they're seeing something more than normal, are there any diseases, any trends? Nothing. I didn't get any feedback in terms of anything out of the ordinary. For swine, we are seeing the usual suspects for winter, which are mainly influenza and PRRS, keep us busy every winter. Perhaps in the last month, a little bit more PED, that would be porcine epidemic diarrhea virus. We see it every winter. It's a disease that hit us really hard in 2013, 2014, but then it got under control. But every winter we see some cases, and in the last month we are starting to see some cases. Again, not a whole lot, but there's some PED virus circulating. And something that we are watching is Mycoplasma hypopneumonia. That's a very important pathogen of swine, and more farms are working on eradicating that one. And so we're trying to see if we see a reflection of that on the samples that come through the VDL.
Hey, John, this is Pat Rivers, the DNR calling.
And so far,
Sorry, I missed your call yesterday.
it looks. So I think there's somebody who has the microphone on. So that's for swine. For poultry, again, in 2022 we got fewer cases of disease investigation, and I think that it was in part because of concerns of high-path avian influenza, biosecurity, and so on. So we got less cases to investigate in 2022. But the type of cases that we're getting, I'm being told they are similar to what we usually get. The only thing that's a little unusual, and it's just a few cases, so I don't want to make a big deal out of it, but we've seen some poults with neurological disease, some cases where encephalomyelitis, others we're still investigating. So there's a little bit of that. And again, something to keep an eye on and keep investigating, keep looking at that, because it might be nothing, but it might be some sort of trend. What else in terms of testing activity or activity at the labs, at the different sections? We've completed several trainings that are mandatory for our MPTL lab in Wilmar to maintain NPIP status. So we sent somebody for training on salmonella. We're going to send somebody for training on mycoplasma testing. That is mandatory. Some of those trainings have been suspended due to the pandemic, and now a lot of the labs are catching up on those. So we're completing those. And then other things we're doing both in St. Paul and PTL, we are completing our proficiency testing to be able to prove that we are up to date in testing diseases like African swine fever and classical swine fever. So those are tests that get sent to us by the USDA, and we have to prove not just that the lab can perform and can provide the accurate results, but at the technician level, that every technician that we have approved to test those diseases really can pass these tests, and we perform those, we pass those. So we have shown that we are ready to test ASF, CSF, and we actually do every week for surveillance samples. Similarly, we get tests on messaging results, and we've also been passing those proficiency testing for messaging. So that's for testing activity. A couple comments on equipment. If you remember, we got funding from the legislature last year to purchase equipment. That's money that comes to us through the Department of Ag. We set up the contracts, made a wish list, and started ordering. And we're getting to the point where we've told our sections end of this month is a deadline to order things, because we're finding out that some suppliers are having a problem delivering some of the equipment that we want, and we need to have all this equipment purchased and delivered by the end of the fiscal year. So we need to have a final list by the end of this month, order everything, and give a little bit of time for all of these equipment to get delivered and paid for, and so on. So that's exciting. We are ordering equipment in all of our different sections, and it's a lot of work, but it's exciting. And in some areas, we think it's going to increase our efficiency with the new equipment. The other comment I wanted to make on equipment is the university. If you're familiar with our digester, we have a digester that gets rid of a lot of the animal remains that we generate in our lab. And not only that, it's used by many other parts of the university to get rid of their animal remains. This digester is very old and it's causing problems. It's breaking down more often than we would like. But it's not ours. We house it here at the VDL and we manage it, but it really is the university's equipment. And just in the last months, the university has shown a lot of interest in finally finding a replacement for this piece of equipment. And we are in the process of planning what the new digester will look like. It's not an easy project, but the university is, like I said, showing a lot of interest in this and pushing this forward. So that's exciting as well, that we know that in the future, we're going to get a new digester. Finally, let me talk to you about a couple projects that we have going on. There's a couple of tests that we have identified for swine that might be interesting in the future. So we're looking at maybe developing those tests and validating them. So that could help veterinarians, and that could help bring additional testing to our lab. That's one of the projects that I wanted to mention. The other one is we're working on a new online submission form. So most of our clients submit samples to us with paper submission forms. And we do have an online submission form and an online way of submitting samples, providing the information for the samples that people submit. But it's an old system and not a lot of people use it. So we got money from the Farm Bill to develop a new online submission form. And that's a project that was moving very slowly last year, and we've ramped it up. And basically now we are making a lot of process. I am tired and hungry as well. progress, excuse me, on that online submission form development. So that's exciting. And finally, one of the things that I usually don't talk very much about is our teaching responsibilities. One of the things that we do in terms of teaching is we train the DVM students in performing necropsies. So we have a necropsy rotation where every student that comes, that goes out of this college gets training on necropsy at our lab. And we're preparing for an increased class with the students that are going to come with the 2+2 program with South Dakota. So in 2024, we're going to have more students to train on necropsies. And so we're rethinking this necropsy rotation. This gives us opportunities to think outside the box and see what this could look like, as opposed to just adding students to the same groups that we're doing now and having more students per instructor. We're trying to think of ways where we can rethink this rotation. So that's exciting as well. All right, that's what I got for an update from the VDL. Are there any questions before lunch?
I had one comment. Dr. Rovira, I was speaking with somebody, a pork producer just recently, and he had mentioned that how impressed he was. He brought some small pigs, nursery pigs up, and Dr. Rosso met him. And by the time that person got home, he already had some preliminary evaluations of what they saw. So at least they could probably initiate something to deal with some of those observations yet that same day. So that was greatly appreciated.
Excellent, excellent. I'll pass it on. It's interesting because we've been actually talking about specifically that. Sometimes the people who deliver the pigs to us, they are in a hurry. They just want to rush back to the farm and they say, just talk to the dog, but we want to actually meet face to face. And very often we learn things that sometimes we don't learn from the veterinarian. And it's for us very interesting to be able to talk face to face with the people who are taking care of the pigs. So I'll pass the comment on to Dr. Russell.
Thank you.
Thank you, Albert, for your update. We are going to go into closed session for lunch. Erin, can you clarify, are board members going to a different room or coming back to this room?
They will go to a different room so that we can call Jim and speak,
Okay.
and then you can come back.
Yep. So Jim, I hope you heard that. We're gonna call you. Jim, we're gonna give you a call on your phone because we're going into closed session.
I did, I heard that.
Okay, perfect. Thank you. We're behind schedule, unfortunately, but between 12:45 and 1, we'll try and start our meeting at that time.
I'm Elaine Hansen. I'm with the Minnesota Pet Breeders Association. And I have also been secretary of the Minnesota Outdoor Heritage Alliance, which some of you know because particularly they're interested in CWD, which is one of the reasons that I'm always here for the whole meeting. Because I have sort of like two constituencies that I'd like to report back to. I was asked to give you some background on the Minnesota Pet Breeders Association and a brief overview. Task force appointed by the board is doing. That's the Companion Animal Advisory Task Force. The Pet Breeders Association was founded in the early 2000s. Sheila and I were just trying to figure out, was it 2004, 2006? Anyway, it was back there before I was involved in these matters. The Pet Breeders Association is a volunteer membership organization. Many of its members are pet breeders. Some of us aren't. I haven't bred pets for over a decade, but I am still showing dachshunds. And there are services and equipment to pet breeders. Some of the pet breeders are the licensed commercial breeders and some of us aren't. Not all commercial licensed breeders are members of the Pet Breeders Association. All are welcome, but it's not it's not a trade organization of commercial breeders. It's open to anyone who breeds pets of any kind. And people who don't breed are welcome too. They let me in. The association provides and has for over a decade an annual 2-day breeder education seminar that always includes presentations and updates from the board, as well as experts in the animal health industries. And that takes place usually, well, in the spring, usually the middle of April, think Tax Day. Generally coincides with our seminar this year. It's on the 20th and 21st, which is a Thursday and Friday. We've been doing that for the public to know that breeders of dogs and cats or any other pets really do care about what they're doing and having the information to do it well. We have experts in pet breeding in all species that are considered companion animals and pets in Minnesota. And this is a free seminar. There's no charge to attend. All of you are welcome if you care to sit in and see what we're up to. It's a really, really great thing. The first time that I was persuaded to skip a show weekend and go to the pet breeder seminar, I sat through the first presentation, excuse me, and I went out to the registration table to Terry Franzen and I said, who do I make the check out to? I was that blown away by the level of expertise that presenters offered and the interest that the members showed in what those presenters had to say. And really opened my eyes to the difference between the substandard breeders that commercial breeders are often accused of being and who they really are. And I say they in the past tense, now it's we. I'm not a commercial breeder, but we're all the same breed, if you will. So things rolled along, and over the years, there have been many, many attempts to get legislation that would adversely control animal breeding, pet breeding in Minnesota. The animal rights crusaders who promoted this wanted to have a program that was costly and burdensome and would make it extremely difficult for anyone to breed pets. And they almost succeeded several times, but with the expertise of pet breeders and the support eventually of the non-commercial, the hobby and show breeders like me, we were able to get to where there was a bill presented that pet breeders could live with. And the details were hammered out in 2014 and the bill passed. There were representatives there of the anti-breeder groups that wanted a different kind of legislation than they were getting, of the Pet Breeders Association, of veterinarians, and consultation from the board staff. During the hearings leading up to that bill passing, I didn't know, and I think most of our members didn't know much about the Board of Animal Health, who you are and what you do, and what kind of relationship we would have if there were a licensing program and it was in the board's hands. But a couple of things happened in the course of those hearings, and since relationships really are built on communication and trust, this was kind of a classic example. At that time, Dr. Bill Hartman was the state vet and executive director of the board. He said that whatever the legislature did with the proposal to have a licensing and inspection program for pet breeders, they should be precise, they should be clear, The board was not interested in trying to administer a program that was ambiguous and open to a great deal of interpretation. We had other support from the staff. Paul Anderson, who's sitting over there, was one of the people who testified at those hearings. And he said, the board is not trying to advise the legislature whether or not to adopt a licensing program, but if you do it, assign the board to be responsible for administering it, because we know how. We've been doing this for decades for species other than dogs and cats. And we know how to monitor and help the producers monitor their well-being and their health. And it should not be in the hands of any special interest group. It should be in the hands of experts like the Board of Animal Health. Well, thank goodness that view prevailed. And here we are. We've had a really good relationship with the board over the years, and it's been built on that kind of communication and trust and candidness. We don't view the board as an enforcer. We view the board as a partner. We're colleagues. The philosophy behind licensing and inspection is that if a breeder doesn't know what they're doing or doesn't know how to do it right, that they'll get some guidance from the board so that they can meet the standards. If they are disinclined to do what's necessary to meet the standards, then in addition to the guidance and encouragement, there's the possibility of progressive enforcement action. We have, we have no idea what the board members were doing during the first part of the lunch recess. And we don't need to know. It's not our business. That's private data. And the public only needs to know if there's an ultimate outcome that results in a violation of a statute. I'm gonna pause for a minute here and see if I can get away from this program. It happens when I talk too much, and I guess I'm talking. We've been on several cooperative ventures with the board. When the animal licensing bill was passed, one of the things it required was that breeders have a veterinary protocol that they followed. It had to be approved by the board, had to be made a matter of record and followed to the letter. There was an advisory group appointed by the board to provide some suggestions and feedback on that protocol. We were there. I was there. There were members of the organizations that didn't want pet breeders to exist. There were veterinarians and there was board staff. The shelters and rescues were represented there. And together we came up with what was acceptable to all as a veterinary protocol, and the board adopted it with possibly some modifications that I no longer remember, but it's been working for all these years. The next cooperative venture that we went into with the board was the Breeder of Excellence program. Too bad Courtney had to step out for the CWD hearing. She was the leader of the program at that time. And, excuse me, the advisory group was comprised of all licensed commercial breeders. There were about a dozen people. I attended most of the meetings as an observer. And that program was developed in keeping with the requirement that had been built into the statute. So we had standards. I'm not weeping, really. That's an effect of whatever it is that's going on in my respiratory system. which is not contagious. Thank goodness. Anyway, over time, things rolled along, everything, the nuts and bolts were working, and it was just humming very agreeably. Concern about the animal rescue industry in Minnesota. Animal shelters have been licensed and regulated since something like 1967 by the board. Did you know that? However, rescues didn't exist per se at the time. They developed over the first decade of this century, and they're now mighty. There are estimated to be 300 to 400 rescue groups in Minnesota that are collecting random source dogs and doing some degree, we hope, of medical and behavioral rehab, and then selling them back to the public. There's no, other than the requirement that every animal coming into Minnesota has to have a CVI, there's no other regulation that pertains to them. And we know for a fact that an awful lot of animals have come in, perhaps fewer more recently, but in the early years of this movement, lots and lots of animals came in with absolutely no health history known at all. Some of them, most of them come domestically from southern states. Some of them come from overseas. And anytime you bring animals in without controls of virtually any kind, and particularly as regards overseas imports, frequently fraudulent credentials, is diseases and parasites introduced into a population that is not familiar with them and not resistant to them. As this industry burgeoned during perhaps the last decade mostly, the Minnesota Veterinary Medical Association took note because their members were treating a lot of owned pets in Minnesota that were showing illnesses and conditions that were not endemic to Minnesota. And these were coming from domestic imports from southern states and from overseas imports. So, what's it been, a year and a half? The MVMA's president, then Connie Sillerud, spoke to the board and made a presentation about these concerns and this issue, and suggested that the board take a position that it should also regulate the rescue industry. Well, Chair Compart said, all those in favor say aye, and bingo, there was a resolution to have staff gathered up recommendations and appointed on behalf of the board representatives of rescues, of shelters, of breeders, of veterinarians, of public agencies, and excuse me, who am I forgetting? There's somebody else in there. And that's where we are today. The task force has been meeting periodically. The necessity to meet virtually has made it difficult at times to convene meetings and get participation. Many of the people on the task force are actively employed. I'm not, I'm retired, so I can pretty much push my own schedule around. But many people have difficulty making time for the task force meetings. Interestingly enough, one of the people on the task force is Dr. Graham Brayshaw, veterinarian for the Animal Humane Society and a former member of the Board of Animal Health. I had the great advantage of serving on one little subcommittee with Graham, and it was just wonderful. He did the heavy lifting, and when I got his recommendations, all I had to do was a tweak here and a quibble there. And bingo, my work was done. So people on the task force, even though we come from different perspectives, there's a lot of like thinking in terms of what we need to do and how to go about it. So we're making some progress. I was hoping that Veronica would have a big presentation today to say, well, here's what we've done, and we've just got these little things to wrap up and we'll be all over with. Well, we won't. It's not that easy. But we're chipping away at it. We'll be recommending, as the board requested, some means to regulate the rescue industry and get a handle on the probability and actually the certainty that certain diseases and parasites that we haven't been seeing much of in Minnesota for decades, but have been showing up in the last few years, are curbed. And the health of the animals coming in and the health of the animals already owned by Minnesotans are protected. So that's essentially where we're at. And if anyone has questions, I'll do what I can to answer them.
Refresh my memory on the task force. Is that over a certain amount of time that task force, and then they have to come up with these recommendations
Yes.
when they sunsets?
Dr. Thompson, before she left us, something that I forgot to do at the beginning is to thank the board for its incredible record of brilliantly choosing a series of state veterinarians and executive directors that is just unmatched anywhere in the country. I network with people all over the country about legislative matters, and what we've got here is the envy of just about every other state. So, and the state veterinarians in turn have done an outstanding job of selecting staff. And I've gotten to know not just the staff involved in the licensing program, but by attending your meetings, I've gotten to know what others do and how they go about it, and about some of the laws and the science behind it. And that's worth coming to St. Cloud to sit all day at a meeting. It truly is. And so we're grateful to have this kind of partnership. Dr. Thompson sent me a battery of information from the statutes and state rules about the appointment of task force. And as best I can recall, if an agency like the board appoints a task force, it has a lifespan of 2 years, but it can be renewed. And the board has to set forth what it wants the task force to do. The task force doesn't have an independent authority, but can make recommendations to the board. Excuse me. There's a cap of 15 members, as I recall, and probably some more details that are escaping me. Dean, do you remember anything else?
2 years, that's the stuff that I remember, and then it does sunset, and then I think if they renew, I'm not sure if you have to tap into another group of people, or if you can continue with the same people that are on the committee or not.
I can't remember that part either. It's addressed. I just don't know which way it goes.
Yeah.
So we're looking forward as the Pet Breeders Association to expanding our membership to more pet owners and pet breeders, and to continuing a really good partnership with the board. We look on each other, I believe, as colleagues. You're not enforcers. That's not how you see your relationship with us. And we appreciate that. We respect that. So we're the animal breeding experts when it comes to dogs and cats, as well as members of your staff. But as far as the public goes, we're the best game in town, and we're very, very glad to be able to work with you in that capacity. Thanks.
Elaine, thank you for that. That was really a good oversight of that program. I don't think since I've been on the board, but I've heard that. So thank you very much for your time.
Okay.
You're most welcome. Do we have Stephan online? Is she available?
Yes. How are you? Uh-oh, can you guys hear me?
We can hear you. Can you hear us?
Okay, yes, I can hear you now. And I will go ahead and share my presentation. Apologies, everything's moving slowly. Okay, can you guys see that?
Yes.
All right, great. Thank you all so much. Thank you, Chair Sawatzke and board members for the invitation to provide USDA updates. I also want to thank Scott Fier and Brenda Hartkopf for allowing me. I've had an opportunity to visit their operations in the past, so thank you all for that. I apologize that I can't be there in person today, or that I wasn't there in person today. For the 6 years I've been AVIC, I also do the vet student interviews with the College of Veterinary Medicine. And so I had a interview that I had signed up for prior to knowing the dates for this, and had that overlap. So it's something I really enjoy doing, helping to pick the next flock of veterinarians, right? So my first update for today is related to new CWD herd certification sampling options. In late December of 2022, USDA released a new guidance titled Sampling Options for CWD Herd Certification Plan. And this was to replace a missed mortality or poor quality sample submission. As you all know, to be enrolled in the CWD herd certification program, owners must conduct CWD sampling on all deaths of all cervids aged 12 months or older. These are deaths that occur on farm, at slaughter, or at hunt facilities under the same ownership. Occasionally, owners with herds in the CWD HCP program have missed mortalities or submitted poor quality samples. In response to this issue, APHIS developed a live animal testing options that state animal health officials can use in white-tailed deer herds when CWD samples are missed or of poor quality. For that approval, the ABIC and SAHO review the previous 3 years of inspection reports and testing history. The owner has to be in compliance, meaning 5% or less missed or poor quality samples. Then if the SAHO and the ABIC agree to this testing deviation, then they put that in writing that the herd would meet the criteria for this sampling option. Another thing I wanted to highlight in this new guidance is sampling strategies for herds that know their genotype versus ones that don't. If the genotype is not known, for one missed sample or poor quality sample, 5 white-tailed deer between the ages of 2 and 4 years have to be sampled. And so between the ages of 2 and 4, and have the longest exposure to the white-tailed deer with the missed mortality or poor quality sample. If the genotype is known, you have those, those criteria, along with owner providing the genetic documentation, and then sampling a total of, at least 4 out of the 5. So you still sample 5 animals, but at least 4 of those have to be at have to be 9.6 GG, excuse me. And if there's no GGs, then you have to test 2 GSs. This sample collection is done by either a state or federal VMO or accredited veterinarian under the supervision of a state or federal VMO. And those samples have to be submitted within 7 days of collection. And these samples are covered by the owner. So these aren't regulatory sample types, which is in line with live animal testing prior to now. The next update I have is related to traceability. So the ADT proposed rule that we've been waiting on was published in the Federal Registry on January 19th. And so now there's a 60-day open comment period. The primary change to the rule was that the requirement that official identification tags for cattle and bison be readable both visually and electronically. And so that means that they have to be readable 30 inches with 20/20 vision, right? So those of you who still have 20/20 vision, bravo, you have to read it from 30 inches. And so there's no change in the proposed to the type of animals or movements required to have official ID. And then state officials in the sending and receiving state still have the option to agree upon alternate forms of ID. And so that's something that was on the horizon that we've been waiting on. And so, of course, you know, there's still some time after the comment period. Those comments have to each be addressed. And so still some time there. Last item is a quick hiring update. So we still have 4 positions vacant with Minnesota Veterinary Services. I'm happy to say we do have a new veterinary medical officer starting in the Duluth area the end of this month, Dr. Amanda Doran. We're really excited to have her join the team. I still have 4 positions vacant though, an animal ID coordinator position, program assistant, animal health technician, and our epidemiologist position is still vacant, but that announcement is actually out currently and closes on the 13th. And so if you know anyone who might be interested in serving as an epidemiologist, that's the position that was formerly held by Dr. Mary Donahue. And so, yeah, have quite a way. I think between the board and VS, we're doing quite a bit of hiring, or maybe that's everyone, I'm not sure. The other thing, I stuck a little note down there to myself to also mention that USDA has established what they're calling a Ready Response Corps. This is a corps of 12 veterinary positions, or 12 positions for veterinarians across the more affected states for HPAI. These are permanent positions that will be created just to do HPAI work, or I should say avian influenza work. The position description is currently being worked on, and we hope to see the announcement for that position here soon. And so that'll be a newly created position, and it'll be located in one of our poultry dense counties. And so I'm looking forward to adding that person to our team as well. And board members, that is it for my updates.
4 positions, obviously the epidemiologist, I couldn't do that one. Could I do any of the other 3? Are they all veterinary positions?
No, the animal health technician position is not, well, actually, the only veterinarian in those 4 is the epidemiologist. The rest are non-veterinary positions. So yeah, Dean, we'd love to have you. You should send you the link.
Yeah.
I have a question about the CWD testing. Now you say the live testing, you know, we can do the 5 animals or 2 depending on the genotype. How is that being approved when the USDA doesn't have a live test approved? I guess
well, this live animal testing would be done by IHC. and it's remote testing. So immunohistochemistry test. And that would be done at NVSL.
Could you elaborate on that? Just maybe break it down to a farm level.
I'm sorry?
Could you break that down, what you just said, the IHC? Could you break it down to like a farmer level?
Oh gosh. This is where I would use Dr. Donahue. I don't think I can.
Do you need blood or tissue?
Tissue.
IHC.
Correct. It's rectal biopsies.
Okay.
Right.
And that's an approved test or a live test. That is an approved live test, the IHC.
That's IHC.
Correct. Yep. And it has been for a while. So the IHC and these rectal biopsies have been in the program standard since 2018.
So my understanding is you have to be specially certified to collect that sample, and we have one person in the state of Minnesota who can collect that sample right now.
Sure.
And nope, okay, yeah, so I am familiar with this. It is a rectal biopsy. It's got to be collected by someone who's certified to collect it. So we have to get some people trained before we can do this. And what Stephan just described is an option. At this time, it's not a mandate. So it's still, we've talked about it, but I think it's important that we do discuss this and look at the pros and cons.
And this kind of like, we've been looking at the live testing and we're telling, okay, USDA hasn't proved anything. And then all of a sudden we're finding out that there is a test there that is approved. So kind of confused.
So yeah, and so a few things. It's an option. It's not the first option. And so the primary requirement is that you test all deaths. And in rare occasions, if you have a missed sample or a poor quality sample, there's some other steps, right? Because if you have a poor quality sample, then the state vet might say that you need to be retrained or use another sampler. And so this isn't a first option. I do think it's also encouraging, at least for me, because I feel like APHIS continues to move forward with options for the producers. I know being regulatory, we're not moving at the pace that maybe the producers would like us to move, but it's not taking us 10 years to do updates. Like our newest program standards had come out in 2018, and then a revised one came out in 2019, and now we have this additional option for missed or poor quality samples. And so we're I mean, again, I know it's not. I think everyone wants RT quick, things like that. We're not there yet, but we are moving forward. So I think having this option for those missed samples, so that a producer who's been compliant for years and years doesn't lose their level, their certification level is really, I think it's a really good move.
Dr. Schaefbauer and Dr. Garcia, it's maybe something fair to say, and correct me if I'm wrong, but the rectal sampling, that's going to be pretty invasive, right? So that's going to be something sedated. You're going to need someone trained. So that's going to be much more expensive. And just immunohistochemistry, IHC in general, is more expensive because that's staining. You have to have a pathologist look at it. So it's going to be a lot more expensive and in-depth, putting your animals through that and stuff. I would say probably why that hasn't been pushed through as a standard, easy way to screen. It's pretty invasive.
Yeah, and the liability of all of that falls on the owner as well, right? And so if an animal is lost in the process of that testing, I mean, all that falls on the owner. And so you're absolutely right. Thanks for bringing that up.
Yeah. So you notice the difference between what your genotype is as to how many samples you need. And I kind of read the document that Stephan prepared for us, and there is a different sensitivity and specificity depending on what animal you're testing. So that's something else to consider. What if you get a false positive out of this thing? Right? And then the other thing to remember is this is to help you keep your certification, which is obviously tied to exporting. There are some states that 100% testing of your dead animals, period. So even though the federal government says, hey, there's an option, if you want to export to that state, which is a state I know you want to export to, they want 100%. So I do think it's great that USDA is welcoming, new techniques, but there's a lot of others and then type of things.
Right. Right. Thank you, Dr. Garcia.
Are there any other questions for Stephan? All right, thank you for your time, Stephan.
Thank you.
Erik and Shauna.
Sure.
First.
Sure. Okay, thanks for having me. Dr. Erik Jopp, the Assistant Director here. I'm just going to give a quick overview of where we are right now. Unfortunately, it hasn't changed a whole lot since our last meeting, so I will go through it quickly, and if there's any questions, I can take those at any time as well. And then Dr. Voss will go into some of the more of the program updates that we have. So here's the epi curve of where we are right now. Day 0 being the end of March of 2022. The red line is this year's outbreak, and the golden line is 2015's. So you can see how differently it's acted from 2015 until now. The green line on the bottom is the daily activity of when cases were occurring. So this is more, depicts that green line on the bottom, the daily cases, since it started back in the end of March. Our most recent case was the beginning of December. The commercial. Can people online see it now? Okay. Yes, our last cases were at the beginning of December. December 7th, I believe, was our last commercial facility. December 9th, the golden was more of a backyard flock. And I believe the last one that was infected, they are doing environmental sampling in the lab today. So that should be our last one to finish out basically this fall surge. Breakdown of what types of birds were infected through the 2022 event, most of them being commercial turkeys. 80 of the 110 facilities were commercial turkeys. What's different with 2022 versus 2015's outbreak, we did not really have any backyard flocks. I believe there was 1 or 2 back in 2015, and this year we had 27. It's acting a little differently than what we saw back in 2015. And this one shows where in the state those cases occurred. The darker the blue, the more cases, so it's by county. It does follow where the commercial birds are for the most part, but there are some outliers with those backyard flocks. We had some in the Twin Cities metro area, and in areas where there was not the commercial presence. Moving on to what it looks like nationwide. This is as of last, the 3rd of February. All of the areas on here that are highlighted are the counties that were affected. So most of this being in the Midwest, but it's border to border. The most recent case on this one is in Pennsylvania. So they've had a few commercial facilities in Pennsylvania that have gone positive as of late. A little bit of a breakdown of the amount of cases by state. As you can see, it's a little hard to see, but Minnesota, we have had 110 cases, 83 in the commercial side of it basically, and 27 backyard. So as you notice here, 748 across the country, we've had over 100 of them. So we've definitely had our fair share. And this is a breakdown by week of the cases across the United States as well. The different color codings are commercial versus backyard. And in the US, it started back in basically this time of the year last year, so beginning of February. So what we know right now, it's an H5N1. I think they're all coming out as this Eurasian lineage. It's a lineage that is highly pathogenic for poultry and extremely transmissible. It does continue to circulate in wild waterfowl, not only waterfowl, but it's worked its way into the higher food chain as well, into the raptors, the eagles, the hawks. We're seeing it in basically everything. I think the last one that I saw come through was in a crow. So it's in multi-species. And we're anticipating it's going to be returning here in the coming months because of it circulating in the wild, as well as when the migration of the wild birds starts coming north. They are finding the virus circulating in those wild populations down in South America as well. So I know it's still in those populations, and as they move north again, we're anticipating that we will see it again. So we do have our ongoing response efforts. We do have our continued disease surveillance. We do have a hotline and a website, the form to report any sick birds. We are, I believe early in the week, we did have one person that had some questions. We contacted them. And if we believe samples need to be taken, we are sending somebody out to take those samples, or in a commercial facility, they would be taking samples by themselves as well. We also are continuing to collaborate with the University of Minnesota Extension with their outreach. Some of the things that we're learning and plans of moving forward, we're going to continue to leverage the existing partnerships that we have and their expertise in the response. We're exploring what responsibilities can be accomplished by others. We're working with accredited veterinarians to do some of the response on positive farm sampling. We're working with producers, industry. So we're looking at, as you've heard throughout the day, that labor is an issue, and this is a very labor intensive process. So we're trying to streamline it as much as we can and make it as easy on everybody as possible, but we still have to respond appropriately. USDA is changing some of the regulations of what they're requiring of us. A little bit less paperwork. There are several different things that we're implementing moving forward because of what they've changed, and we're anticipating that that's going to continue as well as this outbreak continues across the country. With that, I take any questions.
As the board, so since we anticipate that it's going to come back this spring, I mean, we have to go into spring with that mentality, right? So are we doing anything differently compared to last year?
It's a little hard for me to comment since I wasn't on the board last year to see exactly what the preparations were.
Sure.
We do still have our incident management team activated. We do meet weekly. Dr. Garcia and a couple other people have meetings set up this week yet with some industry veterinarians. We're continuing the outreach through extension, those types of things. And then seeing what we can do to respond. And, we still have everything set up as, so if something comes in the lab positive today, we're ready to go by morning on those. So we have those things still ready to go in place. And then biosecurity and all of those other things on the producer side is basically what our best response is right now.
Right. So like our equipment, our depop equipment is maintained and ready to go, and we have inventory on foam. I mean, even those sorts of details.
Yes, we have a maintenance schedule on all the foaming equipment, and that's being updated on quarterly, if not more. There was work done on that, I know, within the last 2 weeks. On the equipment, our foam inventory, we have a baseline that we need to have at least 40 barrels of foam on hand at any given time. We believe right now we have 44. So those inventories are being monitored. They're being.
And the most important thing is porta potties, and yes, we still have some.
When you gotta go, you gotta go.
You'd be amazed how much time we talk about porta potties. They are there. No, so those are great questions. So I know Erik is gonna have a meeting with the case managers and talk through what went well, what didn't go so well, what can we do slightly differently. We're looking at other states. I've had conversations with other states about how they handled it. We use a lot of resources here in Minnesota, a lot. And are there ways that maybe other states have found with fewer resources and still as effective? And can we learn from those lessons? One small example is we typically send a case manager and a site manager. Other states, for large companies, think of Genio, Select Genetics, will only send a case manager and let the company handle the site manager business under an oversight of the case manager. That's one less person per depot. That's pretty good. Right? That adds up pretty quick. So we're looking at that. I do have a meeting with the poultry veterinarians on Friday, and that's all, layers, broilers, everybody. Carol Cardona is lining that up for me. And then I spoke to Dr. Tedrow in South Dakota. As you know, a lot of turkeys, a lot of positive flocks.
Yeah.
Slightly different. They've got the Hutterite communities there, right? So they have got a lot of labor, which is an important part of it. It's the people, but what they found was these little, and he sent me a PowerPoint, they're like the size of a 5-gallon bucket, and they're a foamer. And 2 of them equals 1 Kifka. Just imagine the difference in size of equipment and junk to haul around from farm to farm. They're $1,100 each. But you need the labor, right? So, but that's one of the things that we're going to talk, that I've already talked to Ashley Coles at MTGA. I showed her the PowerPoint. We've invited Dr. Tedrow to speak to the industry on the 17th. So we're looking at ways to be more efficient and take lessons learned from the entire country.
On a positive farm, how many kilos do you have? I mean, I know that will vary.
It depends, I think of a Genio with a bunch of different barns, right? So yeah, it really depends. Yeah, that's right.
Any other questions for Erik?
Any idea how, as far as the breeder flocks and that type of thing, was there abnormally large number impacted, which is impacting availability of poults and so forth downstream, or any comment or anything along those lines?
I know there were some breeder flocks involved, and how that works throughout their industry, I don't necessarily know. We don't see that type of stuff, but.
Yeah, Dean, so I'd say at least on the turkey side, the breeder companies have their birds spread out pretty well across the country and even internationally, with the exact point of let's be able to spread the risk. Yeah, there's some specialty breeds, sure, but.
Thank you.
Quick introduction, I'm Shauna Voss, Senior Veterinarian with the Board of Animal Health in charge of poultry programs. I know I missed the introduction this morning, so just make sure you guys know who I am. I was gonna give you a quick update on just the normal day-to-day poultry program activities that we do because it's not actually all about HPAI. And I think we've gone a number of years without providing some of those updates. And so I just wanted to kind of touch base. And first mention the Minnesota Poultry Testing Lab. Dr. Rovira mentioned that as well in his updates. It is a joint venture between the Board of Animal Health and the University of Minnesota. So we've got all of the testing functions that happen out at the lab, our university functions, and those are university staff. And then the board maintains its poultry program staff out at the MPTL so that we're working together on the day-to-day operations and can provide that kind of one-stop shop for poultry producers. As far as testing, the University of Minnesota is our official laboratory for the board, and all of our board program testing and National Poultry Improvement Test Program, National Poultry Improvement Plan testing happens out at the MPTL. In addition to that, they're available for diagnostic testing that's billable to the client, including bacteriology, serology, molecular testing, and then necropsies. The Minnesota Board of Animal Health is the official state agency for the Minnesota Poultry Testing Lab, for the NPIP, and that is located out at the Minnesota Poultry Testing Laboratory. It's currently myself and 3 staff located at the MPTL. To touch a little bit about what we do, within our rules, you'll see reference throughout this PowerPoint, the rules that apply to this is we authorize poultry testing agents. And so these are people out of the farm. I think Erik is a testing agent with us to collect samples. And so per board rules, it says if you're going to collect samples for board or plan program requirements, you have to be an testing agent. You have to attend a board approved training program, which we offer throughout the year. We just did a backyard gear testing agent course a couple weeks ago, and I've actually got a commercial course scheduled for tomorrow. Once you attend that training course, they used to be in person. We have been doing them virtually for the last couple of years, which has worked out pretty well. And then we follow up with field school training. So then I work with our field staff to go out and meet one-on-one with these producers to kind of get that hands-on practice and training. For the commercial side of things, I have the producers and the people who are working to become a testing agent send samples to the laboratory for us to check kind of quality control on those to make sure that they're collecting good quality samples, that they know how to fill out a submission form. That authorization is then valid for 4 years, and every 4 years then we'll schedule a reauthorization training where they don't have to attend the same course, but we'll meet with them out in the field, at their location to provide updates on the board and anything that's changed over the last few years. We currently have close to 900 testing agents within the state that we certify, and that's both backyard and commercial. One of the benefits of having people trained to do this, certainly when we think about HPAI, is the ability to have people on the farm trained to collect those samples. So I make sure that every single commercial testing agent knows how to collect an oropharyngeal swab for avian influenza testing. That's just something that we want to make sure that they do, even if it's not part of their normal NPIP testing expectations. To give just an overview of the NPIP programs and where that kind of overlaps with our board rules. When you look at NPIP, it's divided up into 2 parts. So there's the part 145, which focuses on breeder flocks, and then 146 focuses on our commercial birds. So in Minnesota, under our breeder flocks, we have our board rules kind of synchronized to match what the NPIP programs have for their requirements. So we've currently got 232 poultry dealers within our state. They fall under subpart E, which is kind of a backyard category for NPIP. And then as far as our hatcheries and breeding flocks, we've got a little over 100 backyard, or we call them WEGB, waterfowl, exhibition, game bird, and backyard flocks that participate within NPIP. We've got 15 upland game bird producers, and that falls under a relatively new subpart within NPIP called subpart J. We've got 1 rat type producer, and that's somebody who owns emus, and they breed, and they've got a breeding pair. And then we've got 69 commercial breeding flocks, and that's both commercial breeder table egg chickens, commercial broiler breeders, and then commercial turkey breeders. And then 8 commercial hatcheries within the state. If we look at 146, part 146, which is the commercial side of things, there's not a requirement within our state rules that says you have to participate in the NPIP with this, okay? So this is a completely voluntary program, but at least on the commercial side of things, the industry knows the value of having participation, and so almost all of the very large commercial operations are participating. So we've got 6 slaughter plants that are participating within the NPIP, and with the slaughter plants, then they have all of their affiliated flocks that send birds to those plants that are actually doing the testing in order to certify the plant. And then we've got 29 table egg layer flocks. So these are just your commercial chickens that lay eggs for human consumption.
Shauna, can you just say what NPIP stands for?
So NPIP is the National Poultry Improvement Plan. And I think at the last board meeting, Dr. Hayes talked about SHIP, which is the swine health improvement that's kind of spun off from the NPIP. One of the other things that we spend a lot of time doing, and it's something that came out of HPAI 2015, was the NPIP biosecurity principles. And I know we've talked about it at board meetings in the past, but it's been a number of years. And so just to kind of remind you guys again, after 2015 and that huge devastation, you know, there was a need recognized to get some sort of basic biosecurity principles incorporated into the NPIP. And provide that kind of minimum standards that producers could follow. And at the General Conference Committee and the biennial conference in 2016, a set of biosecurity principles was put together and voted on. And those principles were approved and became effective in July 5th of 2017. So we're going on about 5 years, 6 years, sorry, of these NPIP principles. And these other principles, and I don't need to go into any of the details, but there's 14 principles that go anywhere from just the training and review of the program all the way to the programs and protocols that happen on farm. But the last item that I've got highlighted here is auditing. And that states that the official state agency, which in Minnesota is the board, has to audit the biosecurity plans, the written plans, every 2 years. And so that's something that we've been working on. The audit process itself is paper-based. So we ask that the producers submit a paper copy of their plan along with training records, maps of their farms, just so that we can kind of evaluate the protocol against the program standards and to the best of our knowledge and ability to assess if it's being implemented on site. For companies and producers with multiple farms that all follow the same plan, we have them submit one plan, and then we'll audit and kind of target a handful of premises within that operation to make sure that that seems to be followed on the individual farm. And the reason why this is important is because when we look at high path response and low path response on the appraisal and an indemnity, so this is actually a clip from the Appendix 2A, which deals with the appraisal and an indemnity. It says at the time of the outbreak, there was, to the best of our knowledge, a biosecurity plan that was being followed that should have prevented the introduction of high path. And there's actually a spot to put a date of that last audit done. We've got about 100 facilities or biosecurity plans that we have to audit every 2 years. Okay, last year, 2021, I should say that was almost 2 years now, we audited 44 plans, and we've got about 55 that were being audited in 2022. Because of high path, because our auditors are also case managers, and just the busyness to that, we are still working through our 2022 biosecurity plans, audits and reviews. NPIP has over the years allowed kind of a grace period, up to 6 months to kind of get those done. And so we will get those audited and approved because I understand the importance of making sure that these producers get the indemnity that's owed to them. And that's all I had for a real quick update on poultry programs, if you guys have any questions.
As far as I know, for 2015, once it was behind us, you know, they stepped back, kind of took a what do you call it, a hot wash, I think Dale called it. Has there been a chance to do anything like this with this one this fall, or because this thing is just kind of ongoing, there really hasn't been an opportunity to look at any differences between now and then? Is there anything along those lines at all?
I'll probably turn that question over to Dr. Garcia or Dr. Jopp.
Yeah, there was a hot wash that was done after the spring surge, and we have not taken the time to do one after this fall.
But you're getting with the case managers.
Yes, yes, but I'll be working more on an individual basis with the hot wash we did. Over this past summer, we brought in bigger groups, and with this one, I'll be working a little bit smaller groups moving forward.
Any other questions for Shauna? Thank you, Shauna. We have Carol ready, Carol Cardona.
Hello, all. Hello?
Can you hear us?
I can hear you, yes.
We can
I'm
hear
going to
you.
share my screen here quickly. Okay, so I'm going to talk about HPAI in the poultry industry. Can you see my screen?
Yes.
Okay. So I heard Erik talk about the outbreak of 2022, and I'm going to give you a slightly different look at it. You know me, always have a unique twist. So this is a national view of what the HPAI outbreak looked like for the calendar year of 2022. And I think you can see that clearly turkeys are, outnumbered in terms of the impact on that species. But we can also see that the backyard flocks had two surges, clearly in the spring and in the fall. But at a 30,000 foot level, you can see that in the spring, we had heavier impact on commercial birds. And I think, some of that, I'm going to argue, is related to, I think, the better preparedness of the industry in the fall. So our Mississippi
Hi,
flyway
Carol, it's
looks.
Michael. Sorry to interrupt. Your screen that you're showing is the PowerPoint window, not the presentation view.
Okay. Let's see if I can get it to the presentation view. As my friend Marie Culhane would say, Carol, you've been doing this for 2 years this way, so come on, right? I'm going to show you my entire screen.
Stephan had her hand up. Is that maybe what she was going to say too?
I'm sorry.
Yes.
Okay, now can you see my presentation?
Yes.
Okay, good. Okay, so in the Mississippi flyway, you can see that things look a little bit different. But one of the things I want to point out here is that the southern part of the Mississippi flyway, and this is true across all the flyways, had significantly fewer outbreaks among commercial birds and fewer among backyard flocks. So it's quite interesting because I think those are two separate surveillance systems that might tell us something. So I think it's important to look at who was infected in 2022. And as you can see, 30% of the cases across the nation were turkey cases, while 58% were backyards. So I think it tells us that turkeys are, again, highly susceptible, and birds that don't have biosecurity protections, i.e. small flocks generally, are susceptible. And that tells us that I think there's some opportunities there to change how things go. So this is the most common question I'm asked, which is, when will this go away? So the virus is here to stay. This virus comes from the goose Guangdong lineage discovered in 1996. They are in North America to stay, either through two mechanisms, either because they will continue to circulate in North American species of wild birds, or because new strains will be introduced annually for the foreseeable future. That's because, you know, we saw multiple introductions of this strain of HPAI virus in wild birds coming to the United States, this time through Europe. In 2015, it was, you know, through Asia. So I think it's here to stay. So will this go away? I'm going to tell you, I don't think, this is my opinion, that cases have to occur. And they certainly don't have to occur at the rate that they occurred in 2015 or in 2022. So cases occur when HPAI moves between farms. That was a lesson from 2015. And the rapid application of control or stamping out can end that farm-to-farm spread or limit it. And that's what I think you've seen in 2022, where you had, you know, you had more than 100 cases in commercial poultry in 2015 and fewer than 100 cases in 2022. So good news, right? Cases of HPAI occur when the virus enters farms from wild birds or the environment. We don't know exactly what biosecurity measure it is that does work, but there is evidence that biosecurity itself can work. And what's that evidence? Systems that had HPAI in 2015 had fewer cases or sometimes no cases in 2022. And there were systems in 2022 that had no cases despite being in outbreak areas. So I think that all is good news. You know, it certainly needs work, but it's good news. And then there are some cases in the industry where avian influenza spread was stopped between barns on a farm. So our unit of measure at, you know, at a regulatory level, because of trade, is the farm. So we can't officially say that that's something that happens, but I think it does happen, and it's something that could be exploited in the future. So vaccination is another thing I want to mention, because it can be used to decrease the transmission of infectious agents between birds, or between flocks, or between farms, or between species. Whether or not we vaccinate, there's associated trade disruptions that may occur with some countries if we choose to vaccinate. And that has been one of the major reasons that the United States hasn't vaccinated, or specific commodities haven't vaccinated, because you're all tied together. And the decision to vaccinate is made at a national level because the trade impacts would be national. So how would vaccination change transmission, or how could it? So when we talk about transmission, we often talk about R0. So this article from JAMA shows what the R0 is. The R0 basically is if there's one infectious animal or one infectious human, as shown here, how many additional humans are infected by that one infectious person. So this depends on contact rate, and it depends on what the characteristics of the virus are. So for Zika virus, highly transmissible, the R0 is between 3 and 6.6. So that means that a single infectious person would infect 3 people or 6.6 people, depending on, you know, sort of the contact rate. Measles, the most highly contagious here, has an R0 between 11 and 18. Ebola, an R0 of 2, for example. So let's see what the R0 is for HPAI in 2022. For broilers, it was 5.5. So each individual broiler that was infected 5.5 additional birds. So this is a fast-moving virus. So it's up here in the Zika category. What about for layers? It was even higher at 9.5. And I'm sure you can guess that turkeys were even higher at a whopping 17.2. So this is work coming from the Secure Food System team, where we model time of introduction. I just want to sort of conclude this part of the talk with saying that the world has changed. We aren't going to see a return to a world where HPAI is not a threat. But we can work to have fewer cases. We can work to figure out what biosecurity works. And we can use vaccines to augment that protection. We can reduce those R naught numbers by doing these things and thus having fewer cases. So can the response be improved? You know, I've heard you all asking that, and that, you know, of course, the Board of Animal Health is there giving you, you know, you guys are all getting the right answers. So here is what I'm going to say at a global level. The simple answer is someday, maybe. But our trading partners are told what we do in response to HPAI. So the significant changes, you know, Dr. Garcia is making innovative changes that do shift personnel that you have available. But to make huge changes, we'd really have to do that, you know, through and still adhere to those trading obligations for the nation. However, we do have better tools available. And so those old trade agreements sometimes lock us into responses that aren't necessarily necessary. So for example, we are better at rapid detection, quick depopulation, and containment. So precision stamping out, not killing an entire farm's worth of birds, perhaps is something that we should look at. Perhaps we only have to stamp out one barn and figure out how to protect the rest of the barns. Now that's a little bit further away, but something that we really should consider because we've seen the impact on supply chain from this particular outbreak. So in the future, you know, we have to acknowledge that the rate of change has shifted. HPAI outbreaks are happening more frequently. Prior to 2015, the last major HPAI outbreak in the United States was 1983. And now, a mere 7 years later, we had 2022. And we're expecting a year after that, we're going to have another one. So this rate of change has definitely shifted. And the poultry industry cannot withstand changes that require massive changes to infrastructure and expenditures year after year. They have to have a predictable rate of change to be able to, you know, economically survive. And we have to find a way to live in this new reality. So my thoughts are on next steps, make a plan to transition to this new reality. And our goal should be decreasing case numbers, finding some innovative ways to decrease case numbers, allow the mechanisms of response that we have in place to currently work. In other words, if we dramatically increase the case numbers, as happened in 2015 and 2022, where you saw this, you know, tenfold increase over what anybody predicted with HPAI cases, it was impossible to deal with. So if we can keep those case numbers contained, then the current capacity that we have can work. Improve the response process. It needs to be less traumatic for everyone. And I hear, you know, Dr. Garcia's administration is making some changes that I hope are gonna help with that. It's incredibly important that we maintain the skilled people that we have. We know that over time and with experience, people improve. And so we can't risk burnout, either among the private sector or in the public sector. And then change the rules for control. Let's make the case for stopping HPAI on a farm and update the rules so that can happen. Let's also get vaccination in place. So in my last slide here, I just want to acknowledge that Dr. Kumar, a poultry veterinarian of longstanding and who was a member of the board, he once said, "I wish viruses were the size of ping pong balls so that people could see them." With that, I'm happy to take your questions.
You mentioned vaccination in your very last few statements as something you think needs to happen. And it's going to be a while, maybe to get the world to see that that's a better way to control this in the domestic population.
So I think this is there's a lot of political arguments that need to happen around that. I think the science is quite clear. I was part of an October meeting in Paris where we had an international consortium talking about the science of vaccination. And, you know, With vaccination, we can reduce the spread to new species. In other words, reduce the threat to humans. We can improve the ability for to keep supply chains supplied. We can reduce spread. The issue is how do we do surveillance? So some countries, because frankly, the United States, we started this whole war on for trade, but we said, you know, if you're going to vaccinate, that's evidence that you have high PathAI. So we're going to block trade with you unless you can prove you don't have any virus. And so we think that those are outmoded thought processes, and that's not something we could ever prove. You can never prove that you don't have something that you don't have. We can only say there's a very low probability that you have it. So we need to start talking about reducing the risk or establishing the risk of a vaccinated population entering the food chain or moving from this place to that place is negligible risk, as opposed to having zero virus. Zero virus is not an achievable or relevant measure.
Here, I've got a quick question. So it seems like in the past, it really got carried through the waterfowl and didn't really impact them, but it seemed like it had such more of an impact on the wild species and the raptor, you know, where it was fatal to them. So do you will continue to get more virulent? Because it doesn't seem like doesn't seem very beneficial to that virus to be getting more virulent that way. So
Right. Those are great questions. And I just would remind you, you know, a lot of times when we think about COVID, we think about all of these same questions and R naughts and stuff. But let's remember the host for COVID is how many species again? Oh, that's right. One, right? Humans are all one species. We have 9,000 avian species worldwide, 9,000. They don't all react the same way, just as I am not going to react the same way if I'm exposed to canine distemper as my dog will. And so you're exactly right. And so learning and figuring out what these various species, what their clinical presentations are going to be is incredibly difficult and very, very confusing. So waterfowl still continue to carry the virus. There's a whole bunch of different life stages that we don't know that much about. And among the waterfowl, there's multiple species. So I think it's, you know, and in fact, geese, you know, we see the word geese and we think, oh, that's one species. No, they're very distantly related, some of them. So I think it's important to remember there's just a lot of diversity among the hosts, and that is going to dictate how they clinically respond, how much virus they shed, how much the virus persists in those populations. And so it's hard for us to tell if there's new reservoirs being established. I think we can always go back to saying that waterfowl are most likely the reservoir, but in North American species, it's really hard to tell.
Thank you.
Any other questions for Carol? Right. I don't see anything. Carol, thanks for your information. We appreciate your time.
Thank you.
All right, Joni, looks like you're ready to go.
Hi, I am ready to go. And I'm going to get us back on time. Can y'all hear me good? Okay. Well, I have to apologize for not being with you. I was looking forward to it, but since I was with around 800 other veterinarians last week, I got a pretty good cold. I didn't want to spread around. So one of the things that I wanted to make sure everyone knew is that we have a new commissioner at the Department of Health, and her name is Dr. Brooke Cunningham. And she started in January, and it's a big job, and it's hard to find a good commissioner. And I think the governor has done a good job. MDH has about 1,400 employees in the Twin Cities area, and we have 7 offices in Greater Minnesota. And of course, the reason we're so big is part of us is regulatory, doing nursing homes and restaurant inspections and pools and whatnot. And part of us does infectious diseases like I do. So she's with us. She's an internal medicine doctor. and she has a PhD in sociology. And if there's anything we learned from COVID, is that we have to understand how people are receiving our messages and what people are thinking more than we do in order to affect behavior change the way we'd like to see it. Because, we can have tons of scary facts, but that does not make people do what we ask them to do. So she's in place, and that's really cool. Now I'm going to skip most of everything I was going to say, and I'm going to move on. Since we spent a lot of today talking about CWD, I thought you might be interested to know what the health department thinks about CWD. And it's a really nuanced message, right? And the reason it's so nuanced is that chronic wasting disease, even though it's been studied for decades, and there's no evidence that CWD has been transmitted to a human, there's still a lot of questions out there, and there's, we just have a general inclination that the health department doesn't want people eating sick animals. So, so we've got no infections ever identified in people. We've not seen natural infections with CWD in domestic animals, including dogs and cats, or livestock, except for deer and elk. But in the laboratory, the species barrier can be breached for many of the prion diseases. And this means that a prion disease that's normally found in one species can be made to infect another species. And for this reason, MDH, we don't recommend that anybody consume meat from a prion-infected animal, period. We don't want people to eat meat from sick animals, period. But, so this is the nuanced part of it. No one's been identified with CWD, but we still don't want you eating it. And I feel very strongly about that. Nobody should be eating prions. So people ask also, what do you guys do to look for the risk or the possibility that CWD could make that species jump into people and infect people? Well, the best we can do is that we do surveillance for other prion diseases in humans, the ones that are normally found in people. And the big one is Creutzfeldt-Jakob disease, or CJD. And about 1 in 1 million people come down with CJD every year. So in Minnesota, that's 5 to 7 people a year get CJD. It's a normal human disease. It's sporadic. There's no real known cause to it. There are prion diseases of humans that are familial and are genetic, but CJD is not one of them. It just happens. So we do surveillance for CJD, which normally occurs in people 65 years of age or older. And so we look for anything unusual, such as clusters of disease, atypical case presentations, unexpected age groups, unusual exposure histories that might indicate a new prion disease in humans. And we've been doing this for decades, and we have not identified any potential cases or clusters of cases, though we haven't done some investigations. So that is, that's why it's a little bit hard to explain what our position is on this. And our recommendations for hunters are the same as DNR's. So do not consume meat from known CWD positive animals, period. And, obviously don't eat sick animals. That's just a general tenet of eating meat. Wear gloves when you dress animals. Don't mix deer up in processing plants. And we recommend, but it's totally just a recommendation, that hunters who want to do it test their animals for CWD. But unfortunately, there's no program for this. This would be at the owner's expense. So does anyone have any questions?
Joni, just for clarification, so you had said that CHG really comes out of nowhere.
CJD,
basically.
yes. Yes.
So is it spontaneous? Did it fit spontaneous, or is spontaneous not the right term?
I think spontaneous is the right word. It's not caused by eating anything or your genetics or any of that. It just happens. And, bovine spongiform encephalopathy, or mad cow disease, that there is a similar prion disease of cattle that was discovered after we kept looking for BSE in cattle that's also sporadic, that's not BSE, but looks a lot like it. So we think that most species probably have a sporadic prion disease that just pops up.
Just to back up, you mentioned the BSE, and I believe in England, was there 290 people that died from BSE?
Something like that. Yes,
So-
and when people get BSE, it's called variant CJD, which I think is the most, confusing name you could give it. So I try not to talk about that. I call it BSE in people, but most people call it variant CJD.
Are there any other questions? Any other questions for Joni? Great. I don't see any. Thanks for your time, Joni.
Thanks for having me.
All right. Last but not least, Dr. Garcia.
Almost all of this has already been said. Well, so first of all, I want to say thanks to Erin for juggling the bazillion changes we made to the schedule all the way up until the morning of. So thank you for that and getting us all here and getting it done on time. And then all the great presenters. I don't know about you, but I learned a lot. Learned a lot about how to grow cervids and a bunch of other stuff. So great conversation, great discussions all around. And I hope this kind of showcased some of the partnerships and cooperations and the way we do business, which is hand in hand with a bunch of other people. So I'm just going to talk about personnel at this point. We do have a new business manager. She came over to us from IBM. Her name is Melissa Jansen, but she's not with us today because she flew off to Hawaii to get married, which I think is a great reason not to be here. She hit the ground running and is just doing great things. So looking forward to more wonderful things out of Melissa. I think you noticed Dr. Courtney Wheeler announced herself as assistant director several times. She did accept the position as assistant director for field operations. That's fairly new. So between Dr. Jopp, myself, and Dr. Wheeler, we're kind of redistributing responsibilities, but we're working through that process right now. But yeah, what a great thing to have her there. Of course, that now means we have to advertise for someone to take over cervids and small ruminants, but that's okay. We've also already announced and met Dr. Katie Cornell, who will be joining us at the end of February, taking over the cattle traceability from Dr. Wheeler and the livestock concentration points from Dr. Haves. And then we have completed interviews for 2 district veterinarians, great pool of applicants. We kind of had a hard time choosing. They were so good, and we've made offers and we're waiting to hear back. And then we are right now scheduling interviews for human resource specialist. Again, a great pool of applicants. Melissa's whittled it down. She selected her panel, and they will start interviewing once she gets back from getting married. And then we also advertised for the emergency manager. That position description went out this week. Okay, so then the next thing we'll have to be looking for is a finance specialist. Marita Bliven has let us know that she's going to retire at the end of this fiscal year. So she's given us some time, and we will be able to get some overlap, someone to make sure that we keep our finances in order. Just to touch on some of the outreach, some of the great stuff your staff here has been doing. I think just about every senior vet on staff gave a talk at the Minnesota Vet Med Association. We also had a panel that was the just-in-time lunch series so that they get a free lunch. So pretty much every vet that was at the meeting showed up for the free lunch. And the panel discussion was Board of Animal Health. We had Joni Scheftel and Stephan Schaefbauer with us as well, just to talk about how we work together and what we do and why it's important to get accredited and remember us as they're out there being vets. We've spoken to the Elk Breeders Association, the Farm Bureau. We've taught accreditation at the university for senior vet students. We gave a talk on how to be a successful county fair manager, and we have taught biosecurity to the state dairy inspector. So reaching a lot of different audiences in a lot of different ways. And that is really all I have that hasn't already been covered by someone else. So barring any questions, it's back to you, Erica.
Okay. I do have one question, unless I missed it, but What about Annie Valgetti's position as a legal, attorney for the board?
Yeah, boy, oh boy, are we understanding how important that is. So my entire business unit is Melissa, who's brand new, and then 2 accountants. So we're deciding now to focus on getting her area filled, and then we'll move on to the others, because the other critical one is that tribal liaison, right? And when we heard from Michelle that had we had a better outreach to the tribes, we may have had a different discussion. So there's a lot of positions that we've got to get filled, but yeah, it's on the list, absolutely. Yeah.
Thank you. Any other questions? Okay, we've got one more thing on the agenda before we leave. So I gotta come up to the front for this thing, it's pretty important. So we have, 2 board members that will be going off the board. Their term has expired in January. Microphone. So the first, board member that we'll recognize is Jim. Jim was appointed by Governor Walz to the board in March of 2020. He has decades of livestock experience and enjoys Minnesota's great outdoors. He's brought both of those lifelong passions into board meetings by sharing his voice and encouraging his peers to think critically about important issues like CWD in Minnesota. Jim, thank you for your service, and we wish you the very best. Enjoy the warm weather in Arizona.
He's muted.
You're on mute, Jim, if you want to say anything, but you don't have to.
Thank you.
You're welcome. And now we're going to move on to Dean. Dean, you've been a true leader on the board for many years. You've represented your industry very well, while also continuing to learn about all other facets that make up the board. It's no secret that with the recent retirements here, we've lost a lot of constitutional knowledge. I would argue that you're part of that loss. You've been a great mentor to me from day one, and I'm sure our fellow board members would also agree. You're leaving some pretty big shoes to fill. We can't thank you enough for your years of service and wish you the very best. Please keep your phone on, because I'm probably gonna call you. Dean, we do have a plaque for you.
Yeah, Ben will be getting it back too.
Okay, so before we adjourn the meeting, we'll have to pick a date for the next meeting, which we generally have in April. So board members, if you know your April schedules. Okay. Wednesday's okay, or do we want to go back to Tuesday? So Wednesday, so 5th, 12th, 19th, or 26th?
Earlier is better.
Right. Field work, right?
Well, the 5th is in the middle of, well, Passover.
When is,
Let's see the 5th as well.
okay. 5th or the 12th, does anybody have preference?
Maybe 12, just because I'll be coming back from Chicago in this second thing. Okay, that's right.
Okay, so next meeting, we'll plan on April 12th. Erin, can we work on a location later?
Mm-hmm.
Okay.
Yep.
Okay, and then just a reminder before we close that, us board members need to stay just for a quick discussion on attending, inspections, which we passed in our bylaws at our last, meeting.
That will have to be postponed because Courtney was gonna lead that discussion.
Oh, sure. Okay, so make sure that you fill out your surveys. from
We'll
Courtney.
figure out that survey so
Okay.
that you guys can actually get to it.
Okay,
So,
perfect. Is there a motion to adjourn the meeting?
I so moved.
It's been motioned and seconded to adjourn the meeting. All in favor, please say aye.
Aye.
All opposed, same sign. Motion carries. Thank you, everybody.